So I suppose we can start.
Yeah.
So firstly,
You're muted, sir.
hello, hello everyone.
Good morning.
Good morning,
Morning.
Nina. And I think there are still several people that are trying to join in. So maybe we can just give everyone 2 or 3 minutes. But thank you very much for taking time to join us for this APAMS first scientific session. And for those who were able to join us yesterday, the plenary, it certainly was very encouraging to see that there was this great desire to really focus on something in which Filipino science or scientists by Filipinos all around the world really creates a network for good and really wanting to reach out and to provide solutions to the challenges that we are faced with today. And yesterday was very encouraging because to really put together a whole lot of people who are really working in the forefront and also in the back end of the challenges of the COVID pandemic, not only in our different countries, but really on a local level was very encouraging. So, you know, the macro scale and also the personal solidarity. I think I just want to quote what Secretary Ernie Pernia said, that, you know, he highlighted that this bayanihan spirit that we have as Filipinos really is something that we should not undervalue. And so this example of doing the PAASE is a good example because as our president, Gisela Concepcion, a month ago threw this challenge to the people who are involved in this, that can we do it? And at the beginning, many people said, this is too challenging and let's not do it. But she threw the gauntlet and said, well, if we are going to be defeatists, we will not be able to go ahead. And so when she sent the invitation to the different chairs for the different sessions, she said, well, do you want to be in it? And so I really felt this sense of solidarity, you know, really to, you know, me as an expat Filipino living here in Melbourne, I said, well, it is worth trying. You know, we might fail, but as long as we fail, but not to give up and try. And so it was such a remarkable session yesterday. And so today we go first up as one of the first parallel sessions. I believe there are 3 parallel sessions happening today. And so I just want to draw your attention to everyone that really the conference theme is the outreach to Philippine regions. So as chairs, we were asked to essentially choose speakers that could represent some of the remarkable work that is being done in the regions. So as you can see, there are 6 speakers, including myself, and everyone is going to talk about something of the essentially the challenges of managing our ecosystems, what are the evolutionary impacts of you know, the challenges of modern living, if you like, on these ecosystems. So both in terrestrial ecosystems and aquatic ecosystems. So in many ways, it's a very large mandate to talk about something like this that's very wide, but I think you will be satisfied to see that the speakers that are talking really express a taste of the variety, if you like, the biodiversity of life and what are our challenges in integrating our understanding as human beings in making sure that we care for the ecosystems that we live in, that we are not separated from them, but we really have to understand with our biological thinking how to manage these so that we do not render this unlivable. So as you can see, we have 6 speakers, and without much more, I think we can start off. And I would like to introduce our first speaker. So our first speaker is Dr. Damasa Makando, known to us as Demi. And Demi is a professor of plant ecology and is the head of the Environmental Biology Division of the Institute of Biological Sciences in UP Los Baños. She did her undergraduate and master's degree in UP Los Baños in agricultural science and completed her doctorate in plant ecology at the University of New England, New South Wales. As an aside, I have to say that I probably am one of the people who has valued Demi's doctorate thesis, because when I did my doctorate, one of the grass species she worked with was very important in the research that I had done. So I had read her papers before I actually got to meet Demi 10 years ago. And I was so pleased that I finally put a face to a name and the work that she had, exemplary work that she had done. So Demi provides leadership to the faculty and manages the various teaching and administrative concerns of the division. And you can see she has quite a lot of research interests, which include land use change, impacts on biodiversity and hydrologic balance, and biodiversity, information systems, climate change, and environmental risk, and a whole lot more. And she is also a reviewer of the International Foundation for Science, Philippine Science High School, and several scientific journals. So I would like to hand over to Demi to give her talk. So over to you, Demi.
Yes, so thank you very much, Dr. Yok, Doronila, for that nice introduction. And maybe I just missed something. I was the division head about 2 years ago. So currently our division head is Dr. Junlit. Yes, right. So I'll now share my screen. Wait, where is it? Hang on, okay, right. Yes, so this one. Right. Is this okay now?
Yes,
or,
yes. Very clear, yes, thank you.
Right, yes. Okay, so again, good morning, everyone, and thank you for finding the time to attend to our session for this APAMS 2020, that is on ecology, evolution, fisheries, and marine science. And I would like to thank Eob for asking me to be part of this session. It's really an honor and my pleasure to share with you what we have been doing in one of our research studies. And of course, I would like to thank Ma'am Giselle Concepcion, our very active, very dynamic president of PAASE, for organizing this very challenging first Zoom conference of the PAASE. Okay, so what I will be presenting to you is a part of our study in the 7 lakes of San Pablo City. So I would like to focus in here the impacts of aquaculture and ecotourism, the anthropogenic activities in the lakes. And I have here a long list of co-authors. So these are part of our research team. So my colleagues at the division, my graduate students, and our other study leaders in the project. Right. Wait, I think I have to do the slideshow. Right. Okay. The next one. Is it right? Looks like it's not working. Sorry. Now, okay, so I'll just do it this way.
Oh, yes, I can see it now, yes.
Okay, right. So this paper that I'm going to present to you is part of our 7 Lakes Assessment and Monitoring Program. This is funded by the Department of Science and Technology, and it is being implemented by the National Research Council of the Philippines. The program is composed of 4 projects, and ours is the 4th one. We are doing modeling, finding the recreational and aquaculture carrying capacity. So it's a 2-year project. So what I'm going to present you are outputs of our first year activities last year. So this one is our study site. So of course, here is the map of the Philippines, and San Pablo City is there in the part of Southern Luzon, that is in Calabarzon region. And in the inset map is the San Pablo City map, and you can see the little blue circles, and the largest of which is Sampaloc Lake, which is right there at the heart of the city. And then you can see the other smaller lakes. And this table is showing you the physical characteristics of the 7 lakes, and we got this from Mendoza et al. 2019. So in terms of elevation, most of the lakes are at low elevation ranging from 80 to 100, 160 meters above sea level. It is only Lake Calibato, the one that is highlighted in red font, that is located at a higher elevation, 1,700 meters above sea level. In terms of surface area, as you have seen in the previous slide, Lake Sampaloc is the biggest, about 1 million square meters, while the rest of the lakes are smaller ones ranging from 220 to around 480,000 square meters. Okay. In terms of depth, there's a very wide range of depth, from very shallow one that's only 7.7 meters, and that is Lake Palakpakim, to a deep one, that is Lake Calibato, that is located at the higher elevation. It has a water depth of 156 meters. All the other lakes range from 20 to 62 meters. In terms of eutrophic condition, Mendoza et al. Classified the 3 lakes of Bunot, Calibato, and Palakpakin as eutrophic lake. So they are more, they are richer in nutrients as compared with Sampaloc, Pandin, Yambo, and the other one, sorry, I cannot see, which are mesotrophic lakes. Okay. In terms of the surface area of the lake that has been covered by fish cages, so the data that's being presented here would be the surface area from 2017 to 2018. The red ones here, I would like to point out that, why are they red? That is because I would like to show you that the surface area covered by these fish cages increased within a span of 1 year. So for example, Lake Bunot, from 13 to about 19%. Calibato from 14 to 22%, and Lake Palakpakin from around 12 to 17%. All the other 4 lakes, Sampaloc, Pandin, Yambo, and Mohicap, demonstrated a decrease in the area of the fish cages. Sampaloc Lake from around 10 to around 8%. So please take note that Laguna Lake Development Authority mandated that the area that should be covered by fish cages should be around 10% only. So Lake Sampaloc is trying to follow that mandate. Pandin and Yambo have very small area that's being occupied by fish cages, around only 2%. Pandin is 1.8, much lesser, and Mohicap is around 6%. With regard to settlement that's living around the lake, Sampaloc is the biggest lake. It is in the heart of the city. That is why it is also surrounded by so many houses, 18,600 population. All the others, Bunot, Calibato, Palakpakin, and Pandin had around 8,000 to 8,500 residents. Mohicap, 3,800, and the smallest one, the fewest residents, would be around Lake Yambo, only 2,000. In terms of uses, so you will notice that all of the lakes are classified as being used for fishery. They just vary in the extent of the area that is being used for fishery. Fishery means aquaculture, fish cages, and of course, open fishing. And 4 lakes, Sampaloc, Pandin, Yambo, and Mohicap, are being used for tourism. Right, okay. So this is just an aerial view of the 3 lakes that are being used for aquaculture: Calibato, Palakpakin, and Bunot. So you will notice, you will see the fish cage structures. No, they are floating around the lake. And then the next slide here would be the 4 lakes that's being used for tourism. So we have Yambo, Pandin, Sampaloc, and Mohicap. So you will notice that the vegetation around Yambo and Pandin, sorry, I get a phone call. Sorry. That Yambo and Pandin are covered by lush vegetation, as well as Mohicap, though part of the area in Mohicap is covered by agricultural area. And Lake Sampaloc here is covered by or surrounded by settlements, houses. Right? It's at the heart of the city. Then the next slide here is data we got from the San Pablo Tourism Office because they have a logbook of the tourists that visit the lakes. And you will notice that Sampaloc is still the most popular among the ecotourism lakes. And of course, the local government unit is really developing lake tourism in Lakes Pandin and Yambo. And they have also quite a share of tourists in the last 4 or 5 years. Okay, so that was for the introduction. And then for the objectives of this study, so we conducted a PRA, participatory rural appraisal activities, to elicit the local ecological knowledge on the historical development of aquaculture and ecotourism in the 7 lakes, as well as the changes in water quality through time. We also did actual sampling of water samples. So that's for the analysis of various water quality parameters and to compare the aquaculture and ecotourism lakes in terms of water quality and also in terms of fish diversity. So as for the methodology, these are some shots for our PRA activities. So we conducted timeline, community resource mapping, and also focus group discussion. So here are some shots of the activities taken when we were conducting the PRA. And this PRA was participated by fish cage operators, fisher folks, and Bantay Lawa volunteers. So each lake, we allocated 1 table, and the people that are sitting in there would be all those participants from that specific lake. We also did actual sampling for fish. So we soaked gill net overnight in the lake from 5 p.m. to 7 a.m. And the following morning, we collected all the fish species that were trapped in the gill net, okay, identified them, measured them, and even weighed them. So these are part of our team doing the fish measurements. And then the picture on the right side would be our team taking or collecting water samples from the lake. So we identified 4 or 5 sampling points. So that would be littoral areas near vegetation and littoral areas near houses. And then we took samples also right in the middle of the lake. That's the pelagic zone. We took samples inside fish cages. And if there is a water outlet, we would also take samples from there. And we analyzed the water for dissolved oxygen, BOD, chlorophyll A, and transparency. As for the results, we start with the timeline. So this is the timeline of aquaculture. So we asked the respondents to try to recall as far as they can remember. And so we started with the 1940s. So from 1940 to 1960, there is no aquaculture yet, but there is fishing, that's open water fishing, okay. Then in 1960, aquaculture, fish pens, fish cages were installed, established in the lakes, Palakpakim and Sampaloc. And so there was a rapid increase of the establishment of these fish cages in Palakpakim until around 2000, and in Sampaloc also just a bit after 2000. In the other lakes, Lake Bunot, it was in the 1970s when aquaculture was introduced, and it increased rapidly also until year 2000. In Lake Calibato and Mohicap, aquaculture was introduced 10 years later. That was in the 1980s. In Mohicap, that was from 1980 to 2000. And then after that, there was a decrease. So you will notice that in Bunot, Mohicap, Palakpakim, and Sampaloc, by the year 2000, the number of fish cages that were in there were dismantled because they have to follow the Laguna Lake Development Authority's mandate that it should be regulated and it should be around 10% only. For Lake Calibato, it's a different story. So you will notice here that it's still increasing up to now. Well, that is because their area is still below 10%, as you have seen in the previous table. For Lakes Pandin and Yambo, okay, through the years, so aquaculture was introduced, but only very few fish cages, 1.8 or 2% only of the surface area. This is the timeline of the number of settlements around the lake. So from 1940 to 1960, there are very few. Actually, in Lake Calibato and in Yambo, there was only 1 house, okay, around the lake. In the others, there are very few. The influx or the number of settlements increased starting from 1960 in Lakes Bunot, Palakpakim, Pandin, and Sampaloc. In Calibato, the increase in settlements started in the 1980s. In Lakes Yambo, there's still very few settlements even up to now. Okay, so you will notice here that in Palakpakim, by year 2000, they regulated the number of settlements because there are really plenty of settlements. So because the barangay is around and it's growing rapidly around Lake Palakpakim. And yeah, this is the heart of our PRA because we wanted to learn, okay, their perception on the water quality. So in all of the 7 lakes, the participants shared that from the 1940, okay, 1940s, they have very good water quality. It said that the water in the lake is clean. They can drink the water. They can use that for cooking, and their surrounding is still very pristine. So all of these good water quality condition, okay, change through time. So for Lakes Bunot, Calibato, it changed starting around 1970. After 1970s, the water quality, they said, started to decrease, okay? In Palakpakim and Sampaloc, the decline in the water quality started much earlier, in the 1960s, okay? Now, in Mohicap, the decline in water quality started in the 1980s, okay? You will notice that for all these 5 lakes, the reason for the decline in the water quality, they say, is due to the establishment of fish pens and fish cages, so aquaculture. For Pandin and Yambo, it was only in the year 2000 when they have noticed that the water quality in their lakes started to decrease, okay? And their main reason is due to influx of tourists and the increase in settlements around the lake. As for the resource mapping, we also asked the participants to map, okay, the resources around and inside the lake. So I chose 2 lakes. One is Lake Palakpakim to represent the aquaculture lakes. So we have 4 scenarios here, 4 maps. Each map is spans for about 2 decades. From 1940 to 1960, so this is the scenario in Lake Palakpakim. They just have fishing boats for open water fishing. There are few houses, so still plenty of trees and vegetation around the lake, okay? The following 2 decades, from 1960 to 1980, you will notice that inside the lake, there's an increase in number of fish cages. There is a decrease in the number of trees, okay? But there's an increase in the number of houses. The third one here on the left bottom, 1981 to 2000, you will notice that there's a further increase in fish cages, in the number of houses, and a much less decrease in the number of fruit trees or other trees or vegetation. And from 2001 to 2019, so this is the scenario now. More fish cages, more houses, much less vegetation around the lake. So this one depicts the historical development, the historical changes in the land use and lake use. The same thing with Lake Pandin. This is the one that we chose to represent the aquaculture lakes. So very strikingly, you will notice that in their illustration from 1940 to 2019, there is still lush vegetation around the lake. And there are also from 1941 to the most recent one, there are only much less houses, and they are located at certain area only. So it's basically in the top left corner here of there. So that's where most of the houses are. And then, so we have few fish cages, and the brown ones would be the bamboo rafts that's being used for tourism activities. Okay, for the water quality results, the first one here is dissolved oxygen. So basically, we compiled all the data for 2 time sampling for August and November 2019 for all the different sampling stations for all the lakes. So basically, ecotourism lakes would have higher dissolved oxygen, significantly higher as compared with aquaculture lakes. By the way, the red dotted line here, 5 ppm, would mean that's the threshold that's being for the dissolved oxygen, 5 ppm. right? And then you will notice that we have 0 and 5 here that would represent the depth of the sampling of the water. 0 means surface water, 5 is 5 meters below the surface. The next slide here is for BOD. So it's the reverse this time. The aquaculture lakes would significantly have higher BOD than the ecotourism lakes. And the red line here is about 7 ppm. So though it's still, the mean is still below, but you will notice that for some of the sampling points in the aquaculture lakes, we have as high as 10 ppm for BOD. So this would be those sampling sites or lakes that would really have maybe higher amount of organic material or excess feeds that are undergoing decomposition. This is for chlorophyll A, okay? So aquaculture lakes would have also higher amount of chlorophyll. They have more algae, okay? So actually in Lake Palakpakim, the color of the water is really yellowish, so yellowish greenish. Especially near or inside the fish cages. So the aquaculture lakes would have higher chlorophyll A as compared with ecotourism. And you could even see some of the data that we could have as much as more than 50 microgram per liter of chlorophyll in some of the sampling areas in some of the aquaculture lakes. For the transparency, ecotourism lakes have significantly clearer water. So we can see our Secchi disk up to about even 4 meters, okay, below the surface. The aquaculture lakes would have around 1.5 or 1.3 meters above sea level. Okay, with regard to fish diversity, so these are some pictures of the fish species that we found in all the 7 lakes. So you have their milkfish, different species of tilapia, and then red devil, okay, flower horn, and then ayungin, the letter O, it's a native species there. Okay. This table here is a checklist of the fish species that were collected in our August sampling in the 7 lakes. We did sampling also in November, but we were not able to complete all the 7 lakes. So we're just showing you the one-time sampling. So, but take note also that even the fish, the local fish cage operators or our resource persons there would say that there is quite a variation in the fish species that they could catch throughout the year. So please take note that this is one-time sampling. All right, so I highlighted in red here the native species. We have the bangus, the Chanus, the Glossogobius, Gliuris, and the ayungin, the one there. And then all the other species, the different species of tilapia, red devil, flower horn, are introduced to species. So for Sampaloc, so mostly they have the tilapia, the introduced species, and the red devil plus ayungin. In Bunot Lake, we have 1, 2, 5, 6 introduced species, different species of tilapia, red devil, flower horn, mixed red and green flower horn, and there's only 1 native species that is still thriving in there at that time, Bunot, ayungin. In Mohicap, it's Chana striata and then tilapia. And flower horn. Sorry, it's Parachromis malaquensis and Chana striata. In Lake Yambo, it still has all the 4 native species. This is the only lake where we found the bangus, the native bangus, the Chanus. And then it has the Glossogobius, Giuris, and then the ayungin. And then all the introduced species, the Oreochromis niloticus, the tilapia, the red tilapia, and the other tilapia species. Lake Pandin has the tilapia species, clarias, and 2 native species, Giuris and the ayungin. Calibato, there was only 1 species, only the Oreochromis niloticus, only the tilapia. Lake Palakpakim, only 2 introduced species, the tilapia and the Carassius. And then in here, I would like to show you the abundance of fish species that were caught. Okay, so strikingly, you will see here the very tall red red bar here, that is the red devil. It's very abundant, more than 200 individuals that were found in Lake Bunot. In Lake Bunot, we also found abundant red tilapia and also the cage Oreochromis niloticus tilapia. And then take note, right somewhere in the middle of the graph, you will see bars there for green and red and green flower horn. These are introduced species, and Josh, one of our research team member who is an expert in fish, said that these species are invasive species. And then at the far right, I would like to show you the native species ayungin. So I'm happy here to show you that it is still thriving and it's quite abundant in 2 of the 5 lakes where it was found. So in summary, these different lakes have a mixture of introduced and native species. Okay, so we also computed for the various diversity indices. Lake Yambo would have the most number of fish species, 8. Calibato has only 1, only the tilapia. Okay, in terms of Shannon-Weiner Diversity Index, the highest would be Lake Yambo. Palakpakim has the highest Shannon Evenness Index, closely followed by Lake Yambo. And for species dominance index, Lake Pandin would have the highest one. And for our recommendation, policymakers and local government units should use these findings, okay, in crafting science-based strategies towards sustainable management of the 7 lakes. And so that's it. Thank you for listening, and thank you for your time.
Thank you very much, Demi. That was a very interesting presentation. We'll leave questions for the end after our first 3 speakers. So without much ado, I would like to now present our next speaker. So this is Dr. Jp Cardell, who is currently a researcher of the Research Institute of Tropical Biology and Pharmacological Biotechnology. And also he's in Cebu Normal University in Cebu City. He completed his PhD in biology from De La Salle University in Manila in 2017. He focused on the systematics, ecology, and ethnobotany of Philippine cinnamon under the supervision of Dr. Neil Aldrin Malyari, President of the Center for Conservation Innovations of the Philippines. His current research interests include, firstly, the understanding of use and value of Philippine spices, the ethnopharmacology of wild edible plants, and urban biodiversity conservation. So together with other Filipino botanists from De La Salle University and Cebu Normal University, his most recent work is in collaboration with the Royal Botanical Garden of Madrid, Spain, where he led a book chapter entitled Philippine Plants in the Last 500 Years, perfect for 2021, so hopefully in the post-COVID era. Ethnobotany and Socio-Cultural Significance in time for the quinticentennial celebration of the first circumnavigation of the world. So I would like to welcome Dr. Jp Cardell to speak to us about his work.
Thank you, Dr. Yob, for that kind introduction. Can you hear me?
Very clearly, yes, to me.
Okay.
Thank you.
Thank you. Good morning, everyone. Good morning, colleagues, friends, my former students, and my professors also who are listening and attending this webinar. So today I'll be talking about Today I'll be talking about habitat preferences, endemicity, and proposed conservation status of cinnamomum in the Philippines. Together with me is my advisor, Dr. Malyari. So the outline of the presentation, I'll start talking about the Philippine cinnamon, the morphological features, brief history, taxonomic problems, and then this will be followed by habitat preferences and endemicity based on herbarium records and species distribution modeling. And then finally, I'll end my presentation in proposing conservation status and threats to cinnamon diversity. Okay, so in the last 5 years, we've been talking to people, not only academics, but also laymen about the Philippine cinnamon. So how much do they know about our cinnamon in the Philippines? So unfortunately, it is quite sad that only like 7 or 8 or 7 students or 7 people that we asked are not actually aware about the Philippine cinnamon, that we have cinnamon in the Philippines. So this kind of reality is actually enough to, I mean, it's actually sufficient to justify that truly there is really a need to pursue this, the understanding of Philippine cinnamon, not only in its taxonomic aspect, but also conservation value, utilization, ecology, and others. So cinnamon has approximately 250 species across Asia, South America, and Australia. And in the Philippines, you have 21 species according to Customs in 1986. So basically, when you look at cinnamon trees, you will see the most distinguishing characteristics being the tri-nerved leaves. So if you would notice in this screen, you would actually see these 3 parallel venation here in the leaf. This is one of the most distinguishing characteristics of cinnamon leaves. It also possesses paniculate inflorescences, flowered with 9 stamens, and fruits seated in a copule. So this is what we usually use in characterizing cinnamon species. So basically, we focus on the leaves, of course. And these are the different leaf forms, and then the different inflorescence type, and then the different fruit shapes, as well as the shape of the cupula, which is a very important distinguishing characteristics of cinnamon. This is an illustration from the study of SAW in 2011, and in my own study, I also prepared similar illustration for the Philippines, but it is not yet published at the moment. So before we proceed to the first objective of the study, allow me first to tell you what have happened in the last 500 years, for example, on Philippine cinnamon. So a lot of people are not aware that the spice trade, which was actually started by the Europeans, is anchored in the idea that they are in search of the spices, particularly in the East Indies, where nutmeg, cloves, for example, as well as cinnamon, pepper are actually found. So from 1492 to 1800s, there has been a series of events in the Philippine history in which cinnamon has been discussed and highlighted, not only in terms of propagation, but also in terms of exploitation of this resource. So, however, the most important thing to ask here now is that what made cinnamon taxonomy difficult to study? Why do we really need to study the Philippine cinnamon? At the moment, there is a kind of understanding about people, for example, being confused about the appearance of cinnamon based on leaf characteristics. So the problem on heterophyly and heteroblast is actually very evident. For example, in the case of heterophyly, different leaf forms on the same plant are observed. For example, in this example, there are actually acuminate leaves, and then you also have ovate leaves, and it's actually in the same twig. Meanwhile, heteroblaste is another problem in which different development of distinct types of juvenile and adult foliage shows different shapes. So if parotaxonomists and taxonomists are actually depending only on the leaf structures, this becomes problematic. Another problem is on the presence of intermediate forms, as well as interspecific similarity and the presence of synonymous species. Also on the aspect of whether a kind of sample is a subspecies or is it actually a cultivar or another congener, for example. So in this illustration, you would see, for instance, that this is actually a type specimen in which Burmanii or Mindanaense is actually being proposed as the name of this specimen. On the right side of the illustration is actually like whether it's Mercadoi or Utili. And taxonomists who studied cinnamon are actually changing their identification of these plants for a particular reason based on morphological characteristics. By the way, in the next slides, I'm showing the different cinnamon species that we have in the Philippines based on their type specimen. For example, there's rupestre nanophyllum, Cinnamomum mercadoi, Ibaloy, which until these days doesn't produce fruits yet. You also have Trichophyllum, Anacardium, and then the famous Cinnamomum cebuense of Cebu. Okay. In the last 200 years or so, scientists and historians have studied cinnamon, and it's actually written in the book of Manuel Blanco, Elmer, Merrill, as well as Santos in 1930, in which he also studied leaf allometry. In this case, even in the 1930s, the idea about the confusing leaf morphology of cinnamon is already apparent and understood. For example, you will see here the different leaf shape of cinnamon species in the Philippines. This is Zelanicum, Mindanaense, Mercadoi, and Iners. So with this conflicting ideas and differences, as well as taxonomic problems on the identification, Andrew Kosterman, so he's actually an Indonesian botanist, created a monograph of the Philippine cinnamon in 1986 to aid in the identification as well as in the understanding of the genus by taxonomists as well as botanists. So aside from that, we also have here recent, what they call this one, effort to understand the use, understand taxonomy of cinnamon based on chemosystematics or chemical constituents as proposed by researchers from De La Salle University, in which they compared, for example, the different chemical constituents found in these 7 cinnamomum species. In UP Los Baños, some researchers utilized phenotypic markers like leaf architecture from the research group of Dr. Buot to infer variation, to infer and understand cinnamomum taxonomy based on morphological characteristics on the leaf architecture. In Cebu Normal University, my students also underwent research study in which they also investigated specific species, the Cinnamomum mindanaense, and that despite the differences in the leaf shape, they actually possess a common characteristic, which is the presence of secondary veins that is running diagonal to the primary vein. In the recent years, there has also been a lot of effort for the conservation of cinnamon. So it all started in 1998 with the funding given to Mindanao, and then followed by the NUCA project through the ENR. You also have here the Global Trees Campaign, the Flagship Species Fund from the Flora and Fauna International, the PBSP, focusing on the planting of Cinnamomum cebuense, and then most recently, a project funded by the PTFCF entitled the Pinoy Cinnamon Conservation Project, which started in 2015 and ended in 2018. Most interestingly, the investigation on cinnamomum utilization was actually undertaken by business people led by Home Organic Philippines Plantsville Health in Western Visayas, in which they actually started to produce products out of cinnamon, which is actually a very important step towards making cinnamon known to Filipinos and telling Filipinos about its use and value. So with all these studies, as well as knowledge about the Philippine cinnamon, I was able to figure knowledge gaps which needs to be studied. And one of this is actually habitat suitability and ecological range, among others. So today, my focus is actually to briefly tell the output or the findings of my research on ecology and conservation stats, in which I ask the question, where can we find cinnamon in the Philippines? And can we possibly predict habitats where cinnamon would grow best in the future? As well as on this conservation status, whether the conservation status is actually updated, or is there a need to propose a new conservation status for the Philippine cinnamon? So to be able to answer this question, I utilized species distribution modeling, which is actually a tool that uses computer algorithm like maximum entropy to be able to utilize biophysical and bioclimatic data and utilize this with the occurrence records and interpolate species distribution over a particular geographical area, as well as predict where they would be found in the future. So in my study, we actually employed different environmental layers, as you will see in the screen, and like biophysical variables as well as bioclimatic variables. So all of these are actually available from computer databases. And we utilize this together with quantum GIS to be able to identify areas with high concentration of species. So this flow chart would tell us about how we make use of this world climb data bases, as well as occurrence records found in herbarium records, as well as our current initiative to look for cinnamon species all over the country, and merge all of this together to be able to look to create a map that would actually show policymakers as well as conservation groups where can we possibly find cinnamon in the future, as well as plant them. So briefly, I'm going to share the methods that we employed in pursuing this study. We actually utilized georeferencing method through this methodology adapted from Ramos et al. In 2012, in which we also employ the Philippine Gazetteer, Google Earth Pro version 7, Pickery, and Philippine GIS data to be able to make sure that our occurrence records are actually correct. Because if you would notice, before the Second World War, some botanists would actually just write the name of the mountain, for example, as well as the location in which they collected a particular plant without putting GPS coordinates, which is very problematic if we're going to use this data in modeling. So from this archaic data, as well as the present occurrence records, we inputted them into quantum GIS and Maxent to be able to create this particular map of the distribution, for example, of this species. So another thing that I did on this particular part of my dissertation is that I also reviewed the current conservation status of Philippine cinnamon. And I'm actually happy that with the 5 criteria that are being utilized in the IUCN red listing, at least one is feasible to be generated through SDM. So as long as I have the extent of occurrence, I would be able to at least propose a conservation. The IUCN red list status for a particular species, because recently there has been few effort to investigate the current status of this species based on their number, etc. So this is actually the steps that I did for the computation of extent of occurrence, until such time that I was able to generate this data, which I actually use for proposing the conservation status. So these are the findings of my study. First and foremost, you would see actually this habitat suitability map, and then you would see on the left side panel the species richness, in which you would see, for example, in the map that the darker section of the map or the darker regions in the Philippines would mean that there are actually a lot of species that has been reported or recorded. For example, in this case, you have plenty of occurrence records from Cebu, Bohol, Mindoro, Palawan, as well as in Luzon. We also found that from the different records that we have, the most favorable, I mean, the most favorable habitat is actually located in low elevation areas, in which majority of the cinnamomum species are found. For example, from elevation 0 to 400 meters above sea level. If you would notice here, it's also quite interesting that Cinnamomum bengalensis was actually found in this particular elevation in Mount Apo, sometime in the early 1900s. We also found that among the 26 environmental variables, the three most important that determines or that predicts potential distribution and habitat suitability of the species is, I mean, it includes soil, soil type, land cover, and isothermality. Isothermality is actually temperature evenness throughout the year. That's related actually to temperature. So essentially, these three variables would determine whether the species of cinnamon would thrive or not. So I'm going to show 4 examples of maps. These are actually habitat suitability maps, and you would see that the pink dots here actually the actual occurrence records, and then the colored section of the maps would show the habitat suitability here as indicated by the legend on the left. So you would actually see that even if the occurrence record was reported somewhere else, like for example here, in somewhere in Laguna, a possibility that the species anacardium would also be found somewhere on the northernmost part of the Philippines. It's actually interesting for future conservation planning, in which researchers, for example, can figure out whether truly this species would grow in this particular part of the map. Also, in the case of Cinnamomum cebuense, this is actually a narrow endemic species in Cebu. That's why it's called cebuense, but according to habitat suitability maps, it might thrive somewhere in southern Leyte as well as in Bohol, which is actually quite interesting. And this is generally affected by land cover as well as type of soil as well as aspect. I mean, these parameters contribute much to the suitability of the habitat. Interestingly, Cinnamomum mercadoi, the most widespread species in the Philippines and most collected in the Philippines, also found in a lot of areas. So all of these colored sections of the maps are worth investigating in the future. Okay, so we'll proceed now to the distribution, habitat preferences, and conservation status of cinnamon in the Philippines. So this table is an output of my research, in which if you would notice here, for example, the first column are actually the species, and then the second column is divided into 2. This is based on herbarium records, the actual records in herbarium specimen, and this is based on SDM predicted models, the habitat stability model that I just discussed a while ago. And then this column shows the habitat preferences of this particular species, the elevation where they are found, the population trend, whether they are widespread. NA, it means this was not run in the model because the occurrence record is less than 5. So maximum entropy will not actually process occurrence records less than that number. Types of species were generalist and specialist species based on niche position and width. And then these are the current records that we have. So we have the DNR admin order. Yes, actually, there's a latest in 2017, so I have to update this. And then I use in red list status. And then this is my proposed conservation status based on IUCN criteria. So if you would notice, for example, let's take one example here. You would see, for example, that in Cinnamon Mindanaense, there's actually not consistent in terms of in terms of the conservation, the threatened status. Here, for example, I figure out that Mindanao is actually endangered. And meanwhile, Loher is actually data deficient. That's why probably it's not assessed. Something like that. Okay, so basically, most interestingly in this table, the habitat preferences would also guide conservation planners to decide where to possibly invest their money in terms of planting this species. So for those who are interested to know per species, I can actually share my data in the future. So this is actually the output of my research. And also worth noting is the Cinnamomum cebuense, which is actually an island endemic in Cebu. If you would see here, this is based on SDA predicted models, and Cinnamomum cebuensis is likely to be found in northern Cebu, Bohol, southern Leyte, and Biliran Islands. And if you would notice here, it's actually downlisted from critically endangered to become endangered because recently there has been a lot of effort to locate them and find mature trees of this species. Okay, so in summary, this is actually my proposed conservation status of the different Cinnamomum species, and my base is actually the extent of occurrence. So at the moment, there is no critically endangered species. So that's actually quite a good news for conservation planners. So some of the pictures here are actually showing some of the threats to Cinnamomum conservation in the Philippines, because recently there has been increased utilization and exploitation of this species. In fact, this is quite expensive compared to our common laurel leaves. Like for example, in Cebu, the trade is not reported, not actually documented rather, in terms of how much has been harvested and how much recovery does a particular plant needs to recover to be able to replace the lost leaves through coppicing. Another one is actually, of course, the typical Sweden agriculture in areas formerly occupied by cinnamon. Okay, so before I end my presentation, allow me to share to you that the conclusion of this portion of my research goes like this. So I realized, and based on the results of my study, this research may have implication in the resilience of cinnamon species against drought. That is, they may survive in thin soil over dry limestone substrate and anthropogenic disturbances, owing to the fact that many species were predicted to occur in built-up areas planted with perennial or annual crops. Further, the scenarios that I presented a while ago may pose a beneficial impact to the adaptation of widespread generalist cinnamon species, as all economically important crops in the tropics face the threat of climate change and human-induced perturbations. Moreover, my research suggests that botanical exploration should be intensified to revisit provenances of specialist species, such that corresponding mitigation measures could be afforded to save the last remaining germplasms in an appropriate conservation program. As humans continue to invade the forest for a safer, cleaner habitat, and as development in the countryside attempt to hasten land use conversions, the actual portion of suitable habitats previously predicted by SDM model for generalist and specialist species will both reduce and has such a corresponding implication to cinnamon conservation in the future. To better produce a more robust prediction and draw holistic conclusion on the ecology and the future habitat suitability of Philippine cinnamon under a changing climate, we need to collect more data on occurrences of cinnamon species and improve database of occurrence records accessible to all. This will facilitate a more accurate prediction of future habitat suitabilities that could be used to enhance policies and conservation directions for these species. For future researches on species distribution modeling and predicting habitat suitability of endemic trees, it is recommended to test more hypotheses associated to the shifting of geographic distributions and habitat suitability under a defined conditions of climate change. Such climatic scenarios will define further the extent of niche differentiation of currently studied cinnamon and could be used by conservation managers to ensure that these suitable habitats could actually be occupied by the species and prevent further loss in the future. Thank you and good morning.
Thank you so much, Dr. Jp Cardell, for that very, very informative talk on cinnamon. Meron akong alam na joke about cinnamon. Kasi sabi nila, cinnamon daw is the most selfless plant. Alam niyo ba, sir, kung bakit?
Bakit, ma'am?
Kasi daw walang cinnamon ang nabubuhay para sa sarili.
Sarili lang.
Joke nila. Oo. Anyway, that's a really great talk because I think we all really need to know more about the conservation status of these plants that we have para naman alam natin how to take care of them and to preserve them and to maintain its sustainability in the future. So later on, we'll have the question and answer session for Dr. J and Dr. Demi Makandong. For now, we'll move on to our third speaker. Thank you so much again, Dr. Jp Cardell.
Thank you, ma'am.
For those who just joined us, we are also live on Facebook right now. So hello to everyone who's watching us from the comfort of their own homes. We also have our participants here on Facebook, but we've opened up our research session to other people who weren't able to register to our Zoom session para naman yung mga iba rin malalaman din nila kung ano ang mga pinag-aaralan ng iba't iba nating mananaliksik sa Philippines. All right, so let's move on to our third speaker for today, which I have the honor to introduce. Dr. Agustin Doronila is a senior analytical chemist at the School of Chemistry of the University of Melbourne. He migrated with his family to Melbourne, Australia 45 years ago, where he completed his tertiary education. He has been involved in research and implementation of ecological restoration projects in mining areas since 1988 in Western Australia, Victoria, New South Wales, and Northern Territory. In 2001, he undertook his doctoral studies at the School of Botany of the University of Melbourne on phytoremediation of arsenic-rich gold mine tailings. As a research fellow with the Environmental and Analytical Chemistry Research Group at the School of Chemistry, he has published papers on heavy metal bioavailability and transfer, in particular of arsenic, mercury, cadmium, and zinc, metal hyperaccumulation in plants, food chain transfer of toxic elements, soil chemistry, and plant nutrition. He has also done work on restoration ecology and post-mining reclamation. From 2009 through the Balik Scientist program, he has been involved in collaborations with Filipino researchers and academics, which he will talk about today. So ladies and gentlemen, without further ado, here is Dr. Doronila.
Thank you very much, Jem. Can you hear me all right?
Yes.
Okay, so this is an opportunity for me to share of the work that I have done in my experience as a Balik scientist over the last 10 years. It's good to look back and to really appreciate these collaborations between us in different parts of the world as Filipinos to really look at the betterment and care of our local environments. So my interests are plants that are quite unique. These are plants that live on metal-rich soils, and in the Philippines we have a lot of those. So I just want to put things into context. So what are metallophytes? So plants that, as I said, live in metal-rich soils, and they have adapted and are tolerant to elevated levels of heavy metals in soil. Oops, excuse me. Well, just to put also to an understanding, metals are important in plant nutrients if they are present in low concentrations. A lot of biochemical processes in a plant are co-mediated by copper, nickel, and zinc and iron, for example. And they're usually in very trace concentrations of usually 1 ppm, 1 microgram per gram. So if you like, that is 1 atom in a glass of water. But in high concentrations, metals are very toxic. So I just want to show you some of the key processes that are involved in a way in which metals can be taken up by a plant. So if you look at the bottom of that diagram, metal is acquired by a plant through its roots, because the plant roots secrete organic acids, for example, which essentially dissolve the metals, and so the roots of the plants are able to absorb it and take it up. And then if you look at the left-hand side where I have chelation, what happens is these organic molecules that are present in the plant essentially wrap around the metal, and then they transport it up to the leaf tissues. So the benefit of this is it minimizes the toxic effect of an incoming metal, because metals, ionic metals, produce oxidative stress. So what happens once the metals are transferred by translocation, if we go to number 3, it is accumulated in the plant's tissue and in what are called cellular vacuoles. So if you like, they're little balloons inside plant tissues where the metal in liquid form is locked up, and essentially with all these organic ligands. However, this is only in a very few species, because if you look at number 4, we have what is called phytostabilization, whereby most of the metals that are present in the soil are just stuck to the surface of the roots. So they're stabilized, and most plants that, if you like, live in heavy metal concentrations that are toxic do not take it up. So in one case with some of these plant species, they are very rare. Some species are what we call hyperaccumulators. These are what we can also colloquially call metal-eating species. And these are extremely rare. They only, to date, we have only discovered 550 plant species of flowering plants and a handful of non-flowering plants, which can accumulate metals, or hyperaccumulate in this case. And so these species are known to hyperaccumulate metals such as nickel, which is the most common one, copper, cadmium, zinc, manganese, lead, and thallium, and the metalloids such as arsenic, selenium, and antimony. And it is very rare that a plant will accumulate more than 1 metal or a semi-metal. In the ferns, you can actually hyperaccumulate arsenic and antimony, for example. And it is very curious that most species that can hyperaccumulate nickel, which is the most common metal that can be hyperaccumulated, belong to the cabbage family, the Brassicaceae. But don't worry, because in the Philippines, for example, there are very few of these species. Most of these Brassicaceae are actually herbaceous or soft-tissued plants that live in the northern hemisphere. So, oops, something happened. Excuse me. So what has this all got to do with the Philippines? I just want to take you on a little bit of a historical trip. So in the early 70s, a group of scientists from New Zealand, from the Massey University, led by Professor Robert Brooks, decided to do an important geobotanical survey of plants from the Southeast Asian region. Well, Robert Brooks and his colleagues had the hypothesis saying that, well, if plants grow on metal-rich soils or nickel-rich soils, which are present in the Philippines, so as you can see that diagram, the Philippines, Indonesia, Sabah, and New Guinea have very high concentrations of nickel-rich soils, or the ultramafics. Ultramafic means these are soils which have very high iron and magnesium, and very low in calcium. And so this is a challenge in terms of plants growing on relatively toxic conditions. So Robert Brooks requested pieces of plant samples from herbarium from all around the world, mainly from Holland and the United States, which had collections from these places, and they analyzed them for the presence of metals. And so this is what happened. So the first report of Filipino plants showing heavy metal accumulation was demonstrated by Robert Brooks, and they showed 2 species of Flacourtiaceae, Homalium bittingensis and Homalium panayanum, which had elevated concentrations of nickel. These are not hyperaccumulators, but they're higher than usual, because anything above 100 ppm of nickel usually in a plant tissue signifies that the plant is being poisoned. However, what was exciting was the discovery of a tree species called Renare Bengalensis. This is a tree violet, which is quite well distributed along Southeast Asia and Australia, and also in the Andaman Islands just off India. And they discovered that this species of tree violet actually had concentrations of nickel which are really off the planet. They were more than 1%. And if you see there in the Philippines, Robert Brooks was really amazed that the Filipino species had more than 10,000 ppm, 1% nickel. And this is really quite remarkable. So what happened then after these late 70s, there were efforts to do some field studies in the Philippines. So in 1986, an English expedition studied the vegetation of the ultramafics of Mount Bloomfield in Palawan. So Mount Bloomfield is the mountain that you see when you go to the Puerto Princesa underground river. So from Sabang, the mountain on the background is Mount Bloomfield. So it was led by the ecologists John Proctor and Alan Baker. So Alan Baker was my PhD supervisor when I undertook my PhD studies 20 years ago. And so this is where I was introduced to the amazing world of Filipino hyperaccumulating species. So that's the view from up the top of Mount Bloomfield. I had the opportunity to go there in January 2019, retrace Alan Baker's footsteps and see for myself what it actually looked like and where some of these ultramafic species of hyperaccumulators are. So Baker and Proctor were able to discover and study the ecosystems, and they were able to identify 4 new hyperaccumulators. This was the first discovery in the Pacific, really. And so they discovered Euphorbiaceae, Ameliaceae, and Ocnaceae, and Dicapetaliaceae. And if you look at those pictures there, if you go there now and retrace the footsteps, you will still see the remnants of the mining camp. People were mining for chromite and nickel, and you can see that huge scar on the side of Mount Bloomfield. But what was so exciting about that study was one particular species. So they discovered what they called Philanthus palawanensis, but then they asked a botanist to, to describe it. So the formal description, if you see, was produced 20 years later. But it was so, Baker and Peter Hoffman and Dr. Domingo Madulid, formerly of the Herbarium, National Herbarium, did a formal description of it. And it was dedicated to that man on the green shirt, Max van Balgoy, who was from Sabah. And he is a remarkable botanist. He has an encyclopedic knowledge of most of the flora of Southeast Asia. But so they named that species Philanthus for Max, so Philanthus balguia. What was so remarkable about it, its leaves contained 1.6% nickel, and the stems would also range from 0.5% to about 2 to 3%. And this was easily shown by a chemical indicator called dimethylglyoxime, which if you squeeze, crush some of the plant tissue, it will exude nickel, ionic nickel onto the leaves, and it will produce that quite dark red reaction. So that indicates that it is possibly a nickel hyperaccumulator. So if you cut the stem of that, what really amazed us, what you could literally have 20% nickel in the phloem sap. So it really asks the question, what are the metabolic mechanisms that allow it to tolerate so much nickel? I mean, to put things into context, our cutlery or our watch bands, for example, are usually about 8 to 15% nickel. This plant has liquefied nickel and tolerates putting it in itself. So this is where I come in, because in 2009, I had the opportunity to do a balik scientist under the invitation of the environmental scientists from Ateneo de Manila. So we went to one of these ultramafic regions in Zambales, and because I said, well, let's go there and have a look, see if we can find some nickel hyperaccumulating species. So this is some images of what it looks like. It's a typical ultramafic stunted vegetation with a lot of grasslands. And incidentally, that grass that you see there is quite endemic in the Southern Hemisphere. In Australia, we call that kangaroo grass. Dantonia, sorry, not Dantonia, it slips me at the moment, but I will tell you later. It escapes the name of what kangaroo grass. I think Demi will be able to help me jog my memory. But so what happened is we collected several species, which I had a hunch could be possibly hyperaccumulators. So of those 10, one was expressed in the dimethylglyoxime test, as you see there. I took some samples back to Melbourne, and we did the analytical work, and we demonstrated that it would go over the 1,000 parts per million level of nickel in the dry leaf tissue. So we consider 1,000 ppm as a good operative definition for metal hyperaccumulation. So I told the team in Ateneo, go back there after the end of the rainy season and test these plants again to see whether nickel uptake is much more elevated. So we demonstrated there is much more nickel once the plant is, if you like, in more less dry conditions, if you like. So this was quite fascinating, but it begs the question, and this is a good study that has to be done in looking at how water availability regulates the accumulation of metals. And this is a project waiting to be done. So I just want to point you out to the recent work on localization of nickel by this Brainius Sanua, and this has been completed by, once again, the team in Ateneo de Manila. This requires a lot of intricate sectioning, and so this was done with the aid of light microscopy and electron microscopy. So if you look at those light images, you can see how the distribution of nickel in the cross-section of a leaf tissue. And so we mainly look at the epidermal tissue, which are the ones close to the surface of the leaf. And so you can see those red spots. So the distribution of the red spots through light microscopy indicate that most of the nickel is in the epidermal and also the tissue that just below the epidermis, and there is very little of it in the vascular bundle. So this indicates that it possibly transports the nickel, as I showed in the model earlier on in the schematic diagram. And this was verified with the use of electron microscopy, where the ratio of nickel in the epidermal tissue is nearly 4 times more than the vascular bundle, because it's just transported and then allocated in the epidermis and some of the mesophyll tissue that just under the epidermis. So this is a very good study, which is the very first ultrastructure study of these nickel hyperaccumulators. So now I take you to my meeting with the UP Los Baños scientists. As you can see there in the background, there is the back of Dr. Asuncion Raimundo. She actually rang me up just sort of a few days before I was set to go back home to the Philippines, to Australia. And she said, why don't you come up to UP Los Baños? I want you to talk to us about hyperaccumulators, because there is one of the teams in Los Baños that has just received grant from the DOST to do some work on these metallophytes, but we need a bit more of a background. So I gave a talk there, and I was so pleased with the reception there. What wonderful questions and answers. And so, as you can see there, Edwina was very articulate and very passionate about his questions. So he and Dr. Kimado said, we would like you to work with us and learn about your experience. But they said the only catch was that they had no funding to support my involvement. I said, well, to me, this was an offer too good to refuse. So we wrote up another scientist application, and I was successful to get that. And so that year I was hosted by Dr. Makando in the Institute of Biological Sciences, and then we also worked together with the College of Forestry and Natural Resources. So essentially with the UP Los Baños team, with the forestry team, they had this project to look at the ultramafic areas of the Philippines, look at the ecology of these areas. So they had 4 study sites in Zambales, near where I went to with the Ateneo team, Samar, Palawan, and Surigao del Norte. And this is very important because in these unique rocky outcrops, these are 5% of the land surface of the Philippines. And so this is also very important in trying to understand the ecosystems before they essentially disappear. Because mining nickel mining is done in most of these areas. So very soon after we did that work, we published, and we were able to identify 24 new hyperaccumulated species. So that's the paper there. And I just like to show you some photos of some of the species. Essentially, we come across a nickel accumulating forest. So that is one of the trees there with 1000 ppm. But we also had a very small herbaceous species of Philanthus, just like what Baker and Proctor discovered. But this is a very small one. But it really had these remarkable levels of 1.7%. However, we were excited when Edwino was able to find a new species of tree violet. It's a smaller, it's a tall shrub, small tree, and it is new to science. It was called Renare Nicolifera. And it was also one of these phenomenal hyperaccumulate, which we call hypernickelophores. So that's just the nickel-glyoxine reaction. And so the work that Edwino has done is very important because it helps us understand how to restore large-scale open-cut nickel mines which are distributed all around the Philippines. So the understanding we have done is vital for this, because in many ways it is not difficult to revegetate these areas, because these hyperaccumulators are also very tough species, but all the other species are also very important. So one of the spin-offs from this kind of work is trying to understand the metabolomics, or the metabolic pathways that are involved in nickel hyperaccumulation. So I just want to quickly show you some of the work in progress. So we have started trying to characterize that through chromatographic methods. So if you look there, we've started comparing 4 of the species and look at the different distribution and the amount of the different organic acids, possibly enzymes that can be extracted from these plant tissues. And so we're trying to understand what is important in nickel uptake. And perhaps some of these have some very important potential for pharmaceutical purposes, and also to understand, well, how does a plant tolerate such large issues of oxidative stress? And perhaps we can understand that in looking at some of the other types of degenerative diseases that we are faced as humans. So now I want to go to the work done on arsenic. So this is actually arsenic and copper. So with the Ateneo team, they said, you know, wherever we go, we notice that there are some fern species that are growing in disused and sort of in the mine waste from gold and copper. So up in the mountain province and also south in Mindanao and Surigao. So they found that there was one particular species of fern that was growing very successfully in these places. So what happened was this fern, which is called Teres melanocaulon, was growing in the presence of 2 other known arsenic hyperaccumulators, Teres vittata, which was discovered by our Chinese colleagues in 1998 by Dr. Chow Yang Wei, who worked with me in Melbourne University for 6 months, and he introduced me to these incredible plants. And then also of Pterogramma calomelalos, which was discovered as an arsenic hyperaccumulator by the people from Thailand, from Mahidol University, approximately 18 years ago. So we also had a very active collaboration with these people. But what was interesting is this Teres melanocaulon that the Ateneo team had discovered was prolific, but when we initially tested it, there was very little arsenic present in his tissue. But I said, look, it doesn't mean that it might not be an arsenic hyperaccumulator. We have to learn how to propagate it. And I want to draw your attention to the technical assistant, Mary Jane Apuan, who was an excellent technician, because she was instrumental in making sure that we could effectively propagate, mass propagate the Teres species and actually do the dosing test to see whether it will accumulate arsenic. So if you look at the table above, when you increase the dosing of arsenic, in this case we use sodium arsenate, what happened was the accumulation of arsenic really increased. And so once we get over the threshold of 1,000 ppm of arsenic, then we can demonstrate that this species is an arsenic hyperaccumulator. So very good controlled work. And so we are happy to say that, well, we finally cracked it in the Philippines. We can do some mass production of these arsenic hyperaccumulators. And I think Dr. Claveria will talk about it later on and how we actually do it. So one of the spin-offs is that we can actually think beyond the box, because arsenic in plant tissue could, you know, the Chinese have started doing research where they're looking at the possibility of using this arsenic as a form of treatment for leukemia, for example. So they're doing some very interesting work there. So lastly, I want to go to the work that we have been doing in Melbourne University. When I was in the Philippines, I asked around and said, well, if there are any young faculty or students doing their masters who would be interested in doing a PhD with us in Melbourne University, please talk to me, and we'll see if we can take it forward. So I was happy to say that we had an excellent candidate. So in that picture there, that's a picture of Hannah at her graduation a couple of years ago. And not only did she complete a wonderful doctorate, she was able to meet her husband. And so a wonderful thing is Hannah and Fernando, who comes from Ecuador, Fernando is also an excellent young scientist, have now been gifted with their first child, Rafaela, there. So it was such a joyful opportunity. But now going back to what Hannah actually did. So I asked Hannah, well, are you ready to do something which is very challenging, which is to look at the mechanisms of uptake of mercury and possibly gold in plant species, because there could be some very interesting studies involved in that. Hannah had done her master's looking at the phytotoxicity of mercury in some herbaceous species, so she was well equipped already. So Hannah did her research work on looking at different candidate species, but we were interested particularly in some species which had the potential to accumulate gold. And so we used cassava as our model plant. Well, you might ask, why is cassava very useful? Firstly, cassava is nasty because we know that cassava produces cyanide, and it is through the cyanogenic sugar called linemarine. And so cyanide is released when a plant leaf tissue is crushed, or if you break the roots of the cassava, and the cyanide obviously is produced in high concentrations and be quite toxic. So we thought beyond the box. If it can produce that much, I'm just going to take you to this. Because, well, cyanide is used for extracting gold from low-grade ore in the mining industry by converting gold to water-soluble coordination complex. So what we wanted to show, well, if it produces so much cyanide, does it actually function if we expose it to toxic levels of gold and mercury? So one experiment that Hannah did, where she was looking at identification of the linemarine through cross sections of root tissue. And if you can see there, the red coloring there indicates that the linemarine mechanism is functioning. So in other words, it is predisposed to be able to dissolve gold. So this is very important, and what she demonstrated, I mean, this is a very complex technical method, but she was able to show that the mechanisms are there to extract gold. So she then used an X-ray system called PIXI, proton-induced X-ray emission, to essentially map the distribution of the metals and all other elements that are necessary for functioning of plants. So if you look there, the top G, H, and I, you will notice that when plants actually accumulate, or any tissue that has nanoparticles of gold will turn violet, because nanoparticle gold is not actually gold colored, it's pink or violet. And so in her experiments, already by doing light microscopy, you could already see that possibly there is gold there. And so with the PIXI method, you could see that those yellow spots that are around the epidermis and in the cortex are distributions of gold. So we had to ascertain whether this was real gold. So with X-ray diffraction through our colleagues in Bulgaria, we were able to demonstrate that if you look at those particles there, the more gold that is in solution, in the hydroponic solution, the more gold forms those nanoparticles. So there is an interesting mechanism in the dynamics of formation of these nanoparticles that we have to understand much more. But what was important is we were able to identify that these gold particles are pure gold nanoparticles. They are not amalgam of gold and mercury, which really was quite a remarkable thing. So in other words, in order to accumulate gold, you don't really need to actually spray cyanide into the soil. A plant which is cyanogenic, just like cassava, will do it for you. So this really creates a window of opportunity for some very high cutting edge research, and I think Hannah has pioneered this, and you know, it starts an opportunity for us to take it one step further. So I just want to finish with that, and by summarizing the work that we've been done over the last 10 years. So the Philippines and Brazil are jointly the third major biodiversity hotspots for nickel hyperaccumulators. As of yesterday, I was speaking to Dr. Quemado and Dr. Fernando, and they said that they have identified a new one. So this brings us to a total of 37. Saba, which has been worked on quite a lot over the last 10 years, has come up with 24 species. So in this part of Southeast Asia, we're not very far off in really going to the numbers which have been discovered in Cuba and New Caledonia, where most of the tropical nickel hyperaccumulator species have been discovered. So we have started work on the phytochemistry. You know, the first phytochemistry study on hyperaccumulators has been done in New Zealand from powdered leaf tissue, which was sent there. But now I think we have the capacity to do it in the Philippines. So the first publication of nickel localization in a native hyperaccumulator has been demonstrated. So to now we have 7 peer-reviewed papers on nickel. And then with the arsenic work, we have 6 peer-reviewed papers on arsenic and copper. And then on the work on gold and mercury, we have 4 peer-reviewed papers. So we have a combined output of 17 papers in 7 years. And Dr. Claveria has just notified me that there are 2 more that have just been accepted for publication. So I'm very pleased to say that it's been a wonderful output. You know, this collaboration has been quite remarkable. So I just want to end with that. Thank you very much.
Thank you so much, Dr. Dornila, for that very wonderful and informative talk. I think it's really important for us to have a lot of collaborations with other scientists as well, because that increases our opportunities to, you know, move on and get more funding and be able to work on more things, diba? Ika nga nila, bayanihan really works well. Okay.
Yes. Yes, I suggest that, well, thank you very much for your patience. We've certainly been listening for an hour and a half. So I suggest that we have a 5-minute breather, and then we'll get back to the second half of our session.
Mm-hmm. Are we going to do the Q&A session before,
oh, yes, please.
when we come back? Or you want to do it now?
Well, we might as well do, we can give 5 minutes to Q&A, yes. Thank you,
Okay.
Jen. So you can type your questions if you like.
Mm-hmm.
Or you...
Jem, why is my slide coming out? I can see it. Ah, no, you can see it. It's not seen by people. Anyway. Thank you.
So anyone would like to ask a question? This is our opportunity now. You can unmute yourselves or write into the chat.
Can I raise?
Yes, Demi,
Sure.
please.
Yeah, I think Ma'am AKR has posted a question in our chat room. Okay, for Dr. Makando. Okay, great talk. You have done the study on the 7 lakes. What policy can you recommend to the government for sustainable development? Thank you, Ma'am AKR, for that very good question. That is exactly the objective of our project. So I have shown in the first slide that the title of our project is Recreational and Ecological Carrying Capacity of the 7 Lakes. So on the second year of the project, we are now developing models, ecological carrying capacity model for the lakes. That's why I've sent our analytical hierarchy process, okay, so the AHP questionnaire to Mampes and to Doc Rene Claveria, just and to Jen, for them to answer this questionnaire, because this will somehow help us in computing, developing the index for the various indicators in the model. With regard to the tourism or recreational carrying capacity, one of my students, MS in Environmental Science advisee, Greshel Lynn Perez, has just finished her master's degree this second sem. She will be graduating on August 15, and she worked on the tourism carrying capacity of Lake Pandin. So in that one, she did modeling and did survey and did measurements in the lake. So basically, for that tourism carrying capacity, she was able to compute for the number of tourists per day that should be allowed in the lake to do various activities. So for example, the number of tourists who can do swimming, the number of tourists who can do the rafting and the picnicking, and then the number of tourists who can do the Mom AKR is in there, or walking around the bush. Okay, and then for the ecological carrying capacity, what we are looking in there would be the impact of aquaculture. So the nutrient enrichment in the lake and its impact on aquatic macrophytes and fish diversity, and also on the water quality. So when we have finished this project by the end of this year or early next year, we will be able to have certain policies as to really how, in terms of aquaculture, the number of fish cages and the total amount of nutrients, the excess feeds that should be allowable to go into the lake. Otherwise, more than that, it should be closely monitored by the local government unit. The same thing with the number of tourists that will be allowed in the lakes that they are developing for tourism. Okay, so they should control the number of tourists. And also, I also have another MS student who is looking into microplastics in the lake. So this one is related to the plastics that would come from tourism and also from the residential areas around the lake.
Great,
Okay.
thank you. I think we have a question here for Jay from Vito Butardo. Yes. So Vito says, have there been any comparisons of the sensory attributes of Philippine cinnamon compared with other cinnamon? I was just wondering if our cinnamon is comparable in terms of quality and sensory profile. Jay, would you like to answer that? You have to unmute yourself.
Okay, I think that's a very interesting question. Actually, what has at the present time, that particular research question is still not answered. What the kind of effort that has been explored so far is just to identify contaminants, for example, of the different spices, cinnamon spices, for example, in the market, like how do you call that one? Extenders, for example, and then additives to the cinnamon powder. But in terms of organoleptic properties and like Did I get it right? The question, comparison of like sensor evaluation, for example. I think hindi pa po siya nagagawa so far. I mean, probably in other countries, based on my reviewability literature, but Philippine cinnamon, wala pa kasi number one na problem sa cinnamon sa Pilipinas, yung identification. So only very few species ang medyo, how do you call this one? Certain yung identification niya. So if we're going to compare, for example, if we compare the most aromatic cinnamon in the Philippines, which is Cinnamomum mindanaense, that is believed to be a synonym or of Cinnamomum burmanii of Indonesia. People are suspecting that the Ice Age is actually the reason why those, from Indonesia, passing through Mindanao, yun po yung parang pinaniniwalaan nila why they're actually similar. So I think it's worth comparing the two species because they look similar, morphologically speaking. In terms of organoleptic properties as well as sensor evaluation, yun pa talaga ang hindi nagawa. Pero in terms of chemosystematics, na-compare na po yung ibang species like species ng ibang countries na nakikita din sa Pilipinas as well as other Philippine species.
Okay. Well, Jay, I think you have to talk to Vito because Vito does some very interesting work on metabolomics. So I think there is, and he has a wonderful lab. So nandito rin si Vito in the city of Melbourne in another university. So I think this could be an exciting possibility.
Okay. Okay, thank you, Doc.
Now, I think I'll answer the question from Dr. Lagman. Is the cyanide production by cassava common? How should the general public be advised about this given cassava is commonly consumed? Yes, I have to categorically say that one of the remarkable things about cassava is it produces a lot of cyanide. And the reason being, I think we have understood that cyanide production is a way in which the cassava plant stores nitrogen because it is a very good plant as a poverty crop. So the release of the cyanide actually liberates the nitrogen which the plant uses. But obviously for us who have the potential to eat it, it is very risky. So in other words, we should never give up in trying to inform people that cooking cassava is important because that is the only way to destroy the cyanogenic glucoside, so the release of hydrogen cyanide, in other words. So also fermenting cassava, the cassava leaves or the cassava roots is a very important way of neutralizing and releasing the hydrogen cyanide. So for example, I was amazed to eat laing in Quezon province, which is made out of cassava leaves. But then when I first saw it, I was a bit concerned. I said, I'm sure these people know how to ferment it properly, otherwise they would be dead. But there is no two ways about it. Cassava is a poisonous plant, but really cyanide in this context is very important to know. But people from many parts of the world, in the third world, are very clever in making sure that they don't get poisoned because all it takes, for example, is to eat 5 leaves, the young leaves of cassava to kill a baby. So they just shows you how toxic it is. But, we know that folk knowledge has been very important in maintaining our health eating such a toxic plant. And then there is another question from Vito for me. Has there been any study on the molecular mechanisms of Philippine metal hyperaccumulators? Vito, there's been work on Philanthus balguia from Sabah, and that has been, the metabolomics of that have been done. But these are early days. I think there is space for you to try to enter into this and collaborate, especially with with your friends from UP Los Baños, because you have the technological array to do that. So we can talk about it off air. So another question. Hannah says, release cyanide is also a form of defense against herbivory. So I think, are there any other questions that
Can I ask a question,
yes,
Dr. Yob?
Yes, Mama Kaya, yes, please.
You were discussing a lot about hyperaccumulators.
Mm-hmm.
I've been kept up with bioremediation development in the country. How are they applied, or Is Renee going to discuss that?
I think so, yes. The work in Surigao will be interesting, yes. Yes.
Oh, okay, because the application will be the more the interesting
Yes.
part of all this. Yeah,
Oh,
okay.
certainly. We have these plants that are very tolerant that we can use now.
Okay,
Thank you.
thank you.
Okay. So Renee says, your comments, please. We cannot do much analytical work on hyperaccumulators because we do not have the equipment to do so. It constrains us to understand better these plants. I beg to differ, Renee, because I have discovered, like in UP Diliman in NIGS, they actually have laser ablation where you can do studies on localization of metals in plant tissue. They only use it for rocks, but they can be used for plants. And so I'm trying to encourage people to think beyond the box to use some of these. And then I think most universities have, well, there's probably a Maldi-TOF in the Philippines. And, but you know, what we need is a better funding stream to be able to use this, because you know, you have the germplasm there. Why send it to places like Australia or United States or Japan? You can do it there. I believe you can do it there. Okay. So anyway, I think Jem suggests that we should have a group photo first. So if everyone can please turn on their screens, then someone will take a group photo before we go for a 5-minute break.
I'll count to 3 para everyone is on their videos, and then I'll just press print screen.
Okay, excellent.
Para
Yes.
sa documentation. Do I have everyone? I think I have everyone. Doxy, do you want to turn on your video? So we can see everyone.
Good to see you, Vito.
Yeah.
Vicky, Doc Vicky, are you going to turn yourself on? And Tina, Tina, are you going to turn yourself on?
Ayan si Cathy.
And Girly and
Just
great.
wait.
Oh, there's Hannah.
Everyone
Yes.
to be
Hi,
there.
Dr.
Ayan si Ma'am Chi.
Oh,
Alright,
great.
sige.
Good. Ah, yes.
We can take it again later.
Oh,
But
yes.
I just, kasi baka may mga darating ulit. So
Yes.
we'll just take it now and then.
So we go. Yeah.
All right, 1, 2, 3. Okay.
So
Nice.
let's go back online at 11:55.
Mm-hmm.
Okay.
No, 9:55.
Oh yes, yes. Sorry, I'm looking at my clock. I'm sorry.
It's okay.
Thank you very much.
All right, bye guys.
We'll see you
in
shortly.
5 minutes. There we go.
Okay, I think it's probably time to start. Okay, I think there's still a few people. If people just signify that we're ready to start, just put on chat if you're ready. Okay, so I've got a signal from people. So thank you very much. So I hope you had a good stretch. So now we will go to our second half of our session. Once again, also I'd like to welcome some of the new participants. So now I would like to introduce our next speaker. So our next speaker is also our co-chair, and she's the youngest of the participants. And I'm very proud to say that Jem has been an excellent co-host. And as they always say, if you need someone to deal with technology, you need a digital native. And usually a digital native is someone who is less than 40 years old. And so Jem is someone who's really helped us make this happen. So Jem likens herself to Moana, always coming back to the sea and trying to see how far she'll go with her long-term goal of being at the forefront of marine conservation. She finished a Bachelor of Science majoring in Environmental Science from the Ateneo de Manila University. She was one of 10 Filipinos to receive the prestigious Australian Leadership Award from AusAid, and through that pursued her Masters of Environment degree at the University of Melbourne. Over the past decade, she had extensive experience working for local and international environmental consulting firms, non-profit organizations, and in academia. She's a very avid outdoors woman, traveler and musician. She has gone to the 3 Apos of the Philippines, Mount Apo, Apo Island, and Apo Reef, hiked up some of the highest peaks in Asia like Mount Kinabalu, and gone diving in some of the best sites of the Coral Triangle. She sings with the Bukas Palad Music Ministry, has done local music theater productions, and written heartfelt songs about things she cares about. When she's not working on science, you'll see her out and about playing soccer or training for a triathlon. Today, she'll be talking about her ongoing project, which is part of the Old Dominion University, where she is doing her doctorate under a Fulbright scholarship. So take it away, Jem.
Thank you very much, Doc Yob. Again, my name is Jem Baldissimo. Magandang umaga po sa lahat ng nanonood mula sa Pilipinas. Isang mapagpalayang umaga sa inyong lahat. And to everyone who's watching this in other parts of the world, here in Norfolk, it's already 9:59 in the evening, so good evening to everyone as well. I'm here to talk about an ongoing project that I'm involved in. So the stuff that I'll present to you guys today is more of a preview and an update on what we're actually doing. But the main topic that I'm looking at is at determining the genetic diversity and connectivity of marine ornamental fishes in the Philippines. So before I go through talking about this topic, I want to first zero in on the Philippines. So the Philippines is the center of marine shore fish diversity in the Coral Triangle. But then again, our local marine biodiversity is threatened by things like overfishing, habitat degradation, as well as climate change. And these three factors can potentially make us, instead of a center of marine shorefish biodiversity, a center of marine shorefish adversity. So in connection with this, we tried to find out what the status of the Philippine coral reefs were. So DOST-PCAARD, or the Department of Science and Technology, through the Philippine Council for Agriculture, Aquatic and Natural Resources Research and Development, or DOST-PCAARD, as I've said, funded a National Assessment of Coral Reefs program. And I was actually involved in this project as a research assistant, where we studied from 2014 to 2017 the fisheries livelihood. So in particular, I was part of this project called PEARL, which stands for People and Environment, Assessment of Reef Fish Resiliency and Associated Livelihoods. And in this project, I had the opportunity to go around the Philippines and even go into very remote areas like Tawi-Tawi. And in these areas, we were able to talk to fishermen and their communities and ask them about what type of fish they got, how big these fish were, and what fishing gear they were using. I also had the opportunity through this project to do some fish visual census and see the coral reefs and the fishes up close. And it was actually this opportunity that introduced me to marine ornamental fishing, which is a reef-associated livelihood in some coastal communities. So if we zero in on marine ornamental trade, it does have some benefits. So it provides livelihood for fishermen. It's also a hobby for aquarists, and it provides support for a lot of marine educational programs. So for example, if you are someone who doesn't really have the opportunity to go underwater or go diving, then you'd be able to see this marine life up close if you go to an aquarium or a museum. However, there have been concerns about the marine ornamental trade. It's been found to have a large appetite because of the volume of fishes that are traded annually. And it has been seen to have negative effects. For example, it has been found to be the cause of extirpation and overharvesting in some areas. It has caused changes in trophic compositions in some local reefs. And it has been a cause for introduction of non-native species. So if some of you are familiar with the issue of the lionfish invasions in the Caribbean, they're saying that it was because of an aquarium fish, a lionfish, that was probably flushed in a toilet or just released into the sea. Another concern with regard to marine ornamental trade is the high post-capture mortality, which means that after a fish is caught, they eventually just die out before they even get to the supplier. There has been an underestimated exploitation rate, and it has been associated with some habitat destructive methods because some fishermen use cyanide to stun the fish first so that they'd be able to catch the fish. Now, if we focus only on marine ornamental fishing in the Philippines, not many people would know that we are actually one of the top exporters globally. So the Philippines and Indonesia exports the most number of marine ornamental fishes. The past studies that have been done in our country are mostly regarding import and export trade mechanisms, value chain analysis, and looking at the vulnerability of marine ornamental fishes to overfishing. However, it seems that based on some studies, the effect of the aquarium trade on the coral reef system or the ecosystem itself remains undocumented. There have not been any formal stock assessments, or they would be lacking, and we do have a limited fisheries management strategy in the country. Another finding was that there is a need for understanding the population biology and interconnectedness with other populations. And this is with regard to the targeted species. And it's actually this last point that has further pushed me to work towards a project that can contribute to determining genetic diversity and connectivity of marine ornamental fishes in the Philippines. So going back to our study, the two central questions that we're actually trying to look at in this proposed study is about genetic diversity and genetic flow. So we want to ask, have there been any centennial changes in genetic diversity and in gene flow between populations for ornamental fishes in the Philippines? Now, genetic information like these can actually help add insight to how marine ornamental fish populations have been affected. But you guys might be wondering, how can we actually look at centennial changes? Because that seems to be a very long time ago, right? Well, we have to do a bit of time traveling for that. And so I want to talk about the albatross expedition, which will be the key for us to study centennial changes. Many people know that the Philippines was a colony of the US after the Battle of Manila Bay in 1898. But not many people know that soon after that, the Smithsonian Institution came to the Philippines on the USS Albatross between 1907 to January 1910 to survey the Philippine Islands. And they collected over 79,357 fish specimens. And they preserved this in rum, so the DNA was actually intact. Now, the U.S. National Science Foundation has funded the Philippines PIRE project, which is short for Philippines Partnerships for International Research and Education. This project is a collaboration between governmental and academic institutions in the US, including my university, Old Dominion University, where I'm studying right now, as well as some institutions in the Philippines, which include the Bureau of Fisheries and Aquatic Resources, and the National Fisheries Research and Development Institute, or NFRDI. We also have local collaborators from the Philippines, which are the three universities, Silliman University in Dumaguete, Mindanao State University in Tawi-Tawi, as well as the University of the Philippines in Mindanao. Now, the overall goal of our project is to look into the genetic diversity of the USS Albatross specimens and assess the impact of overfishing and habitat degradation on these species. My proposed project, which will be for my dissertation, shall focus on the subset, which is just the marine ornamental fishes. Now, given these two concepts, the first, which is that genetic variation can infer levels of migration, gene flow, and extinction risk, and the second point, which tells us that exploited populations have a lower heterozygosity, allelic richness, versus exploited populations. Given these 2 points, we can hypothesize that maybe the albatross collections from the 1900s would have higher genetic diversity and higher connectivity compared to the local contemporary collections that we would be getting right now. So we expect these to have a lower genetic diversity and less connectivity. So how do we plan to investigate that? We plan to investigate the genetic diversity and connectivity through going back to the same albatross sites if possible, and then collecting the same marine ornamental species that the USS Albatross collected. And here on the slide, I have some examples of some of the marine ornamental fishes that they were able to collect. So we could either buy them from the market or have some fishermen collect them for us. And then we'd proceed with DNA extraction for both the albatross and their contemporary samples. And then we would genotype them using high throughput sequencing. And then we'll go through bioinformatics where we filter out and we do some data analysis on the genotypes that we were able to sequence, and then we proceed with the population genomic analysis. So what's the status of our project right now? Currently, we've done fish collections in a few localities in the Philippines, in Batangas, in Zambales, for the contemporary collections. Last summer, we went to Camarines Sur, and we were able to collect some fishes there. And we've also done 2 practical computing and bioinformatics workshops. And in these workshops, we held them in Silliman University, where participants were from different universities in the Philippines. And we also had interns who are part of the research experience for undergraduates. And these are students from the 4 collaborating universities from the US. We've also gone through the initial stages of genotyping for some species, and we're also in the initial stages of bioinformatics analysis. Unfortunately, due to the COVID-19 pandemic, our research has been delayed for a bit, but we do have a lab continuity plan at the moment, and we've been trying to adapt to the current situation, and we've looked at things that we can do right now while we are here in the US, and hopefully when it's safe to travel again, we'd be able to go back to the Philippines and continue our sampling. But at the moment, we're all waiting with bated breath if, you know, we would be able to do some genotyping and if we'd be able to do analysis on these specimens from the albatross and the contemporary samples that we have. But I'm really excited to see what we'll find about the marine ornamental fishes between the 1900s and now. So as I've said, since we're still in the process of going through, we're still in the middle of doing our data collection at the moment, you can find out more about our research by checking us out on Facebook and on Twitter. We also have a website that is dedicated to the Philippines Spire Project. If anyone wants to collaborate as well, we are free. Please feel free to reach out to me. I have my email here, and you can also follow my adventures as a PhD student in the US at the moment, a Fulbright scholar, and you can connect with me through social media as well. I'd like to acknowledge the National Science Foundation for funding the Philippines Spire Project, our project collaborators locally and here in the US. I would also like to thank the Carpenter Lab from the Biological Sciences Department of ODU, who has been really instrumental in doing all the field work and lab work that we have here. And of course, the Fulbright Graduate Student Program, who is funding me to be here right now. And lastly, I'd like to thank the Philippine American Academy of Science and Engineering for this opportunity for us to share our current research. We think that it's very important for us to know not only the current science, but also know something about our history. And I was really surprised to know that, you know, the Smithsonian was already established ever since the 1900s, and they even went to the Philippines. Actually, this albatross collection that we're looking at right now is the biggest collection that they do have. And it will be interesting to find something about the science, but also something about, you know, our heritage. The Filipino word for the sea, our sea creatures is yaman dagat, and it's definitely a treasure that we should hold and sustain. And we're hoping that through this project, we'd be able to contribute to that. Thank you very much, everyone. Maraming salamat. And if you have any questions, please feel free to ask later.
Thank you very much, Jem, for that wonderful talk. Very fascinating. And you think, well, when we watch the movie Finding Nemo, you realize, well, the Philippines sends all these Nemos, and your work is very important for that, making sure that we are able to sustain and protect these clownfish. So now I want to present our next speaker. It's Dr. Teresita Perez, who is a faculty of the Department of Environmental Science at Ateneo de Manila. She is also a professorial lecturer in the Institute of Biology, University of the Philippines. So Tess, as we call her, or Doc P, by the students, is a freshwater ecologist where her favorite organisms are the microalgae. She's a phycologist, but aside from the taxonomic aspect, she also studies the ecology of these organisms. Lately, she has worked on metallophytes, considering the country's status being rich in mineral resources, and at the same time a hotspot for biodiversity. Her presentation today is on the riparian ecosystem of the upper Marikina River Basin, a protected area but exposed to anthropogenic pressures. Studies in the riparian areas of this watershed are relevant since a water reservoir dam will be constructed in the midstream part with a hydropower dam in the upstream of the river system. So thank you very much, Tess.
Okay, thank you for the introduction. I will do the slide sharing.
Yep, I can see it.
I'll just move it. Thank you also. It's an honor to be a part of this parallel session sponsored by PAASE. I'm also happy to meet some of my friends attending this parallel session.
Okay.
Yeah, okay. So can you hear me?
Yes.
Yes. Thank you very much. The title of the presentation is Integrated Floral, Faunal, and Soil Assessments of the Riparian Corridors in a Protected Landscape. My co-authors are Brett De Leon, Ruby Descalzo, Eman Lacadin, Rene Claveria, Milet Mendoza, Sofia Dairit, all from the Department of Environmental Science, Ateneo de Manila University, and Francis Magbanwa of the Institute of Biology, UP Diliman. Riparian corridors provide indispensable ecosystem and economic services. Well, as ecologists, you know that an ecotone facilitates dynamic interaction between the adjacent terrestrial and aquatic ecosystem, influencing soil texture, regulating water quality, supporting aquatic life, and enhancing floral and faunal diversity of the area. So this photo would actually show you a part of the riparian area of the river system, where some people would cross from one bank to another. Some people swimming and doing their laundry. Next, this is the figure of the Marikina River Basin, where major municipalities of Rizal are included, together with some cities such as Quezon City, Caloocan, Pasig, Makati, Taguig, and Cainta and Taytay. In 2015, the DNR declared the Upper Marikina River Basin Protected Landscape as a critical watershed, at the same time a protected area. Actually, the original inhabitants of the area are the Dumagats, plus some lowlanders. But eventually, the Aklanans from Panay migrated in the area. So there's actually clans of people from Aklan speaking Ilonggo and Tinaraya when we went there. During dry season, portions of the river is used for local ecotourism. So people would build huts, have it rented out, and families would come over, they will swim and have picnics. And some portions, also during summer season, some portions of the riparian area is converted into vegetable plantations. So the products such as eggplant, tomatoes, are transported downstream via the river and sold to the nearby market. Also, plans are laid down for the construction of a dam in the midstream, as mentioned by Yob, and also a hydropower dam in the upstream of Montalban River. But we need more data. There's a scarcity of studies in this protected landscape. The objective of this study is to provide baseline data on the riparian ecology of the upper Marikina River Basin, particularly Montalban, Boso-Boso, and Tayabasan River. At the center is a photo of the sampling sites. So the yellow pins would show you the yellow, the green, and the red would show you the sampling sites. So this is Montalban River, where the water quality is relatively pristine. And there would be local ecotourism in the area. And this one is Boso River. Water quality is not as good. Effluents coming from backyard piggery as well as hog farms are drained directly into the effluents are drained directly into the river and affecting its water quality. And you have Montalban River, you have Boso River, and would merge into the so-called Tayabasan River. In this part, there's actually a metal bridge that was built by Mapua University. This is a bridge, very sturdy bridge, that would actually provide safe passage or for the community as well as students, because the school is here. So during wet season, the water can really be very high, so that the bridge would actually serve as a safety passage for the people. On the left would show you the one sampling point. The sampling point is divided into 3 zones. Zone A is adjacent to the river. Zone C is adjacent to the terrestrial area, while Zone C is the center. So a belt transect was set up, and in each zone, 3 by 3 meter quadrat was set up also. And this would show you the sampling design. So we study the floral diversity as well as the above ground biomass of the flora. Soil samples were collected from each of the zones, and the fauna, macroinvertebrates particularly, were collected using colored pans. So please take note of this. The soil, on the other hand, were studied considering organic matter, moisture, pH, texture, and nutrients, particularly phosphates and nitrates. This would show you the floral diversity collected from the different sites of the riparian area, 22 families with 59 species. There was abundance and importance of Alphenatera sessilis. And what's important is there's still the survival of the 2 true riparian flora. This is Rotula aquatica and Homonea riparia. These plants are submerged during wet season. But then when summer comes back, the vegetative structures will develop, as well as the flowers and the seeds. So the picture is relatively small. This is actually a ladybug associated with the leaves of Rotula aquatica. So based on our study, all sites have stable to more stable floral community structure. Sampling was done sampling regime was done during the transition between the wet and the dry season. The area actually is basically of the climate type 1 classification, but the upper parts of the basin is classified as the climate 2 classification. Floral zone pairings for the 2 sampling regimes were done using Shannon Diversity Index p-value, as well as the Jacquard Similarity Index in terms of percentage. So based on this analysis, it showed zonation and distinct floral composition among all sites, considering the zones. So this will show you the zones. So this is A, near the river, where the true riparian flora were observed. And this is C, near the banana plantations of the locals in the area, the terrestrial area. And the C, take note of this footpath of the community, so they can move from one barangay to another. The above ground structure, we call AGB, was also studied. So all the biomass in the quadrat were collected together with the woody and herbaceous plants above the soil. So these are samples of the dried biomass were placed in the oven and later weighed. So we observed high species richness but lower biomass. So Braenia cernua was collected. This is actually a nickel hyperaccumulator. The Homonea riparia, as well as Cogon, your Imperata cylindrica, which account for higher AGB in some zones. Generally, no significant difference in AGB, above ground biomass, was observed across zones per sampling area. Vegetation in Site 2, Zone B, is constantly disturbed, as shown earlier in the slide, where this is actually a small footpath for the community. Site B, Zone A, is abundant with talahib. And Site 6 is dominated by herbaceous vegetation. So for the fauna, 12 insect orders were noted. And the top 5 would include your Diptera, you have Araneae, and Coleoptera. There was also the interesting presence of Collembola, which would indicate high nutrient content in the soil. So the high nutrient content in the soil would actually support the floral assemblage in the riparian area of the river systems. All sites have stable to more stable community structure. Zonal pairing was also done using Shannon Diversity Index p-value, as well as the Jacquard Similarity Index, and no distinction or zonation was found considering faunal composition across sites. Flying macroinvertebrates such as butterflies, the ladybugs, the small flying insects were also observed pollinating, because the sampling regime was during the transition between the wet and the dry season, pollinating actually the vegetation with flowers. Very interesting to see. For the soil, this is characterized by the antipolo clay, a fine, mixed, well-drained, with moderate permeability. There was a numerous iron concretions and or weathered rock fragments. Moisture is about 2 to 6%, while organic matter is 1 to 4%. There was a general trend with increased distance from the river. This must be the influence of the terrestrial area. pH is 6.5 to 8.4 average, with a decreasing trend with increased distance from the river, becoming more acidic. And for the nutrients, the phosphate was from 1,400 to 2,500 milligrams per kilogram, while nitrates is 4.5 to 59 milligrams per kilogram. There's no observed trend, and factors that would affect this type of absence of trend would be the varying concentrations of iron, considering the geology of the area, and the aluminum oxides and hydroxides. Texture range from sandy to loamy sand. And there you have slope and disturbance that would influence the range of texture that we observe. So, using the integrated analysis PCA tool, these are some of the observations. 1, generally no distinction among sites for the soil. Soil with higher fractions of clay have higher moisture and organic matter content. Clay particles have greater surface areas, and soil with higher silt fractions have higher nutrient levels. Also, sandy soils hold less water, organic matter, and nutrients as they easily drain. The MDS, NMDS, and CCA was also utilized to observe and study the floral and faunal composition as well as the above ground biomass. There was no observed variation across sites for floral and faunal composition as well as the above ground biomass. This may be attributed to indistinct environmental characteristics of all sites, hence indistinct composition. Yeah, by the way, of course, we use your statistical tools to observe the relationships, but most of these conclusions that we got from the statistical tools have been validated in the field. So for the fauna, We compared the riparian fauna and the benthic macroinvertebrates coming from the river system. There's a significant difference considering the riparian and the benthic community coming from the river. Well, theoretically, the larvae and the pupa of these insects in the river, when they mature, would migrate to the land area, to the riparian area. But considering the sampling regime that we did, which was during the transition, and there was still rain coming in, maybe that was the time for the adult stage of the insect to migrate to the riparian area. Further, for the integrated analysis, you have the NMBS and the CCA tools, and these are the following conclusions and observations. So relationships of certain species orders with soil parameters. So the floral species Cyanodon dactylon and insect order Columbella are associated with the environments of high soil nutrients. It was actually mentioned earlier, and this is very interesting because of the Columbella can actually be an indicator of high nutrient content in the soil in the riparian area. Floral species such as Homonea riparia and Centrosema pubescens and the insect Coleoptera are associated with soil pH. So these are the following conclusions. Compositional zonation exists for flora, but not for fauna. General zonation trends were observed in some soil parameters. I mentioned actually earlier the factors that would affect the differences in terms of the texture as well as the nutrient content in some of the zones and also some of the sampling sites. Riparian and benthic macroinvertebrates are different. So this might be due to the biological and environmental constraints of some species found in the river system, as well as the sampling regime which was done during the transition from the wet to the dry season. Integrated analysis revealed relationships between flora, fauna, and soil parameters, which is very important for a rich and developing riparian ecosystem. All sites, the more stable floral and faunal community for now, in consideration of the existing community in the area. The study sites may be facing disturbances and pressures influencing riparia and flora to change. None of the true riparian flora, the R. aquatica and Escherichia, are in the top important species. They were just found in the zone zone A portion, but was not actually moving towards the other zones of the riparian ecosystem. There was also the presence of too much actually invasive species, Chromolina odorata, Amaranthus spinosum, and Saccharum spontaneum. And there was also the role of the Diptera and the Aranii in the community to enhance the interaction between the flora and the fauna. So for the last slide, these are our recommendations. Improve the uniformity of sampling frequency. Of course, the wet and the dry season should always be considered, although we have to be careful when sampling during the wet season. Increasing the distances between sites with more time for each site. If you're not so familiar with the area, well, I've gone there to check on the dam and the hydropower project as evaluator for the EMB for government. But we didn't have time we just had to work for one day, starting at 9. It was difficult. This area was very difficult to reach. So we had to maximize our time from 9 in the morning up to, let's say, 5 or 6. And we have to go back to the barangay area, considering the road, which is very difficult. Study the two riparian floral species, including their response to severe anthropogenic pressure. There has to be an improved monitoring of this protected landscape and the biodiversity within. Of course, we have to include also the protection of the dumagats, as well as as well as the inhabitants in the area. And the dam and the hydropower endanger an already disturbed landscape. The goal, as conservationists, should be to improve the conditions and not to further destroy it. So thank you very much for your attention and have a nice day.
Thank you very much, Tess. Very important to see studies like these, which really provide the necessary baselines so that we make sensible conservation outcomes. But as you said, there's always a challenge of time, but you know, you've been able to pack it in with a lot of work, and it's really very encouraging that, you know, that there are studies like this. Thank you. So now we go to our last speaker in this session. So we have Dr. Renare Claveria. Renare is an assistant professor at the Department of Environmental Science at Ateneo de Manila University. He is a geologist by profession and has worked in different capacities in government, mining industry, and now in academia. He received his doctorate at the University of the Philippines in economic geology as his field of specialization. His areas of expertise are in mineral exploration, mining geology, petrology, and ore microscopy. I can say that we've managed to make him into a biologist with the association that we've had in the last 10 years. His present researches are on phytoremediation and phytomining, and on trace and rare earth element geochemistry. So thank you very much, Renare. So I think you have to unmute yourself. Renare, are they?
Okay.
Yeah. Okay.
Yeah, thank you, Yob. Good morning. Thank you very much for the very nice introduction. Allow me to share screen now. Sorry.
Yes, I can see your PowerPoint, so you can just maximize it.
Yeah.
Presentation view.
Yeah.
Okay, great. Thank you.
Okay, I got it. Let me move to the first slide. Okay, can everybody see the slides?
Yes, looks good, Doxy.
It's okay. Okay, what I'm going to present now is actually part of a research project on phytotechnologies funded by Fisheries. This is a collaboration among researchers like Dr. Tess Perez, Dr. Agustin Doronila, Dr. Ayan Navarrete, Ms. Sofia Dairit, Mr. Brian Lim, Ms. Elaine Perez, Dr. Dennis Apuan, and Ms. Mary Jean Apuan. The title of this presentation is Recovering Copper and Arsenic from Hyperaccumulator Ferns, Perspectives on Phytomining. Most of us are quite familiar with hyperaccumulators. These are plants that can accumulate metals into their above ground structures at values commonly above 1000 ppm and may even reach to values of about 10,000 ppm. And these plants are not affected by such accumulation. Their values are beyond the normal upper limits of plants in tolerating hyperaccumulated metals. For example, the metal copper. A hyperaccumulator can absorb 10,000, 1,000 ppm or more copper as compared to a normal plant that can absorb copper only to about 30 ppm. Otherwise, the plant would experience phytotoxicity. The general objective of the research is to determine the usefulness of hyperaccumulators. We know that they are very effective in decontaminating the soil of various metals. However, there has been a constant issue on what to do with these plants as they contain relatively high amounts of metals. These plants thrive very well and will always tend to survive in contaminated soils, as these areas are practically their natural habitat. Harvesting and disposing them may even be an added problem. The thought of recovering the metals from the plants would be an interest to most of us. Apparently, this has been done elsewhere. I would like to cite the work of Brooks et al. 1999 on the concept of phytomining, where hyperaccumulators can be used to extract metals from soils that were left behind by conventional mining due to their sub-economic grade. A very good example is in nickeliferous laterite mining, where conventional mining tend to extract laterite or do not tend to extract laterite with grades below 2%, as this is already considered as sub-economical. Hyperaccumulators, as mentioned by Job a while ago, can absorb nickel from these sub-economical soils up to 6%. So in essence, we might as well mine these nickel hyperaccumulators. We made an assessment on the different kinds of hyperaccumulators that can efficiently absorb copper and arsenic, and of which will be used in our metal recovery processes. In Benguet, in a small-scale mining area at Camp 6 along Cannon Road, we noted the abundance of Pterogramma calomilanus, and in an old tailings pond of Benguet Corporation, we noted also the abundance of Pteris vittata. These ferns are well studied and are known to be excellent arsenic hyperaccumulators. We confirm this by their high translocation factor values, indicating higher concentration of arsenic in the shoots and the leaves. Going south in Surigao at Manila Mining, we observed the abundance of a new fern, Pteris milano caulon, thriving very well with Pterogramma calomilanus and Pteris vittata. The copper analysis of the plant showed high bioaccumulation factor values with the high concentration of copper found in the roots, classifying this fern as a phytostabilizer. The arsenic content apparently showed the high translocation factor values, therefore higher concentration of arsenic found in the shoots and the leaves, therefore classifying the fern also as a arsenic hyperaccumulator. It was actually a discovery of a fern that can accumulate both copper and arsenic. After assessing other metallophytes, we took interest on the 3 ferns that can be used for our recovery experiments. These were Pteris milano caulon, Pterogramma calomilanus, and Pteris vittata. For the recovery of copper, we used the roots of Pteris milano caulon due to their high copper concentrations. The recovery is through the electrowinning process. Electrowinning is an old traditional method used in mining and metallurgy for the removal of copper and other metals from the ores. The method is relatively simple, with the use of 2 electrodes, an anode and a cathode, and the application of current. The solution was prepared with the use of nitric acid to digest copper from the roots. Aqua regia was also used, but apparently did not indicate significant differences in dissolution process with that of nitric acid. The experiment setup has aluminum as the insoluble electrode. an acid solution laden with copper, and a variable condition such as voltage and time. From the results of the experiment, the copper in the solution was reduced drastically after the electrowinning process. This is noted at different time frames, 30 minutes, 60, and 90 minutes, as well as different currents, 1.5, 3, and 6 volts. Looking into the percent recovery, there's no significant difference with respect to recoveries ranging from 98.7 to 99%. If we are to assess efficiency, we have to consider the amount of energy used. So the most efficient conditions are with variables 30 minutes with respect to time and a current of 3 volts. For the recovery of arsenic, we use the above ground components of Pteris vittata and Pterogramma calomellanus for their high arsenic content. The recovery process is through the hydrothermal mineralization technique. From previous studies, this method was used to remove metals from wastewater. The concept behind this method is the natural hydrothermal mineralization process. At depths of about 1 kilometer or more, magmatic and magma-heated waters circulate towards the surface. These heated waters are characteristically acidic and can dissolve metals from the surrounding bedrock. In areas where conditions change, such as lowering of temperatures and the increase of pH, with the appropriate conditions, there's a tendency of metals that are dissolved in the heated waters to precipitate and are normally manifested as veins. To mimic this, we use stainless steel vessels with peak liners. The variables to the experiment are pH and temperature. From the equation shown in the slide, to digest arsenic from the plant, nitric acid was used, and in the process have actually oxidized arsenite, As3, to arsenate, As5, which is actually the preferred arsenic species for the recovery process. The formation of arsenate should be consistent throughout the process. In the hydrothermal mineralization technique, a mineralizer is added, that of calcium hydroxide, which would increase eventually the pH of the circulating solution. At lower temperatures, it was expected that arsenic would precipitate as a solid. From the results of the experiment, the arsenic concentrations in the solution were reduced drastically, with the final pH of the solution being more basic. Apparently, arsenic was noted to precipitate as a solid. Though this initial experiment has provided us a possible tool for arsenic recovery, it is recommended that further studies be made. With some successes in the recovery of arsenic and copper, we eventually focused on applying this to ecological restoration and mine rehabilitation. The first thing we did was to propagate the potential hyperaccumulators. We were quite successful in the development of propagating protocols. With the accomplishment of propagating most of the ferns, we eventually built up nurseries to cultivate hyperaccumulators and other metallophytes for various phytoremediation studies and for mine rehabilitation. We grew most of these plants to maturity prior to transplanting them to the rehabilitation areas. We have chosen a particular site at Manila Mining in Surigao, and this is the abandoned Suyok North Copper Gold Open Pit Mine. It is relatively wide, barren area with no vegetation, except for the sporadic growth of few Pterogramma calomellanus. Using this as a reference, we decided to transplant Pterogramma calomellanus from the nursery to this area. The challenge was on plant care management, as we found out that the fern is relatively sensitive to transplanting. Immediate care should be provided regularly, that of water and amendments. If not done, a large percentage of the plant would eventually die. It was really unfortunate that we were not able to do regular monitoring of the rehabilitation site. It is highly recommended that plant care should be provided and in order for us to have a successful rehabilitation activity. The presentation is quite short. I would like to thank Fisheries for funding Phytotechnologies project and the collaboration we had with LaSalle and UP Los Baños on the different related projects. It was a very successful collaboration among the three universities. As a reference, we listed some of the publications coming from the results generated from the research project. If you are interested in some of the papers, please inform us and we are most willing to provide you copies of what you need. Okay, thank you very much, and hoping this presentation has provided you information on the various applications of hyperaccumulators. I would appreciate answering questions if there are any. Thank you very much.
Great. Thank you very much, Renee. Thank you very much, Renee. I thought it was quite fascinating. It was really beyond the box. No, don't worry about the shortness, but I think you have answered a very important question that people always ask: what do you do with the biomass once you've accumulated that metals? But you've demonstrated, and it's proof of concept that you can actually utilize it for some other purposes in really good cleanup. And like I made a comment there, that it really is a great synergy of skills. And thank you very much. So I open the floor to questions, if anyone would like to ask them. And I just wanted to also say, well, I remember the grass species that I slipped my mind when I had my seniors moment. So it's Themeda triandra, or in Australia it's called kangaroo grass, but each country has their own colloquial name for it. But it's a very hardy grass that grows in a lot of these dry, ultramafic areas. So we have 7 minutes before we conclude, so this is a good opportunity. But I think the presentations really indicate that we really have to, we have the opportunity to harness our biodiversity resources in order to face some of these challenges that we have with the way we've tipped things off the scales. But I also want to firstly also say to the co-speakers, thank you very much. We're the guinea pigs of the PAASE parallel sessions, because we were the first ones off. So I'm sure that we've accepted the patience and perseverance of our audience. And I think it's an opportunity to report back to the executive committee and saying, well, sure, we have sort of gone a bit over time, but that doesn't matter. It's not, we're not hampered by the challenges of having to follow a schedule, because we've all been in lockdown. So the opportunity to give space to what everyone has to say, I think is very important. So I'm glad we have Nina Cadiz, who is also chairing a session that I invite people to join later on next week. And she has some very interesting speakers too.
Yo?
Yes?
Can I ask a question?
Yes, you certainly can.
I would like to address this to Dr. Ago.
Mhm.
Ma'am, I know you did a lot of work on transplanting. Maybe you can advise us on the best way how to go about with it.
Just like the advice of Dr. Doronila before, we have to study the succession of species. So it is still good to introduce or to plant the secondary species like the grasses, the sedges, and even we can consider non-indigenous species for the beginning, but we have to manage them well that as time goes by, as the soil is recovering, the general vegetation is recovering, then that's a time for us to plant or introduce the natural species which can actually hyperaccumulate heavy metals. So we just follow the lessons from nature. Start with the secondary species, perhaps from grasses to herbs, and then our more special species like the ones that they discovered, the Philanthus, the Renare, etc.
Thank you. I fully endorse what Dr. Ago said, that we really have to really push ecological thinking in what we do. So I think this is a pervading theme in the work that Dr. Makando and Dr. Perez also show, that unless we have a grip of how ecosystems work, even our proposals and recommendations really will bound to fail, because we oversimplify systems. And so the more we introduce the capacity of an ecosystem to be resilient by adding everything, not just one or two so-called star species, we create monocultures that are always bound to fail. Thank you. So I think Vito has a question here. This is a question to Jim. So Vito says, do you have any backup plan in case we have extended lockdown? I hope you can continue your research on ornamental fishes. It is quite fascinating.
Yes. So I do have a backup plan, but it's not necessarily related to the genetic diversity and connectivity. The thing is, when we try to do these genetic studies, we really have to get samples. So if, let's say, we're not able to get samples, I think one of my backup plans is one of my chapters will be focusing more on just looking at vulnerability of these marine ornamental fish species, and then looking at just establishing the conservation status. There's this idea that I learned, or the concept that I learned when I was still working in UPMSI, where, you know, we can't really know how to take care of something if we don't know what we have left. So if we don't really know the conservation status of these marine ornamental species, then we don't really have a starting point for how we're going to maintain their sustainability. So one backup plan for me is to look at pushing for determining the conservation status of most of these marine ornamental fishes if the DNA stuff doesn't work. Another option that we can do is just to look at contemporary connectivity. So if we're able to establish that, say, for example, you have marine ornamental fishes that you're collecting, let's say, in Batangas, if you can find any DNA connections or connections with their genes between your population in Batangas and then I guess another population in Puerto Galera, then maybe we can say, oh, they can replenish each other. So that's another thing that we can do. So if, let's say, the albatross samples don't work, we were not able to get any DNA from these mummified fish, I think one other option for us is to just look at connectivity between the populations. Okay.
Thank you. Yes. And I think Vito has another question for Renee. Where is it? Where is it? This is a veto to Renee. Renee, what is the future plan for your research? Hope you will continue in this interesting topic.
It depends on the funding, to be honest. So there are a lot of thoughts that we have actually gotten into as a continuance of what we are doing. There are a lot of understanding on uptake mechanisms that we need to cover up before we can actually do more. You have also a lot of plans on rehabilitation. Again, this is constrained by funds. If it's actually provided, it would be very helpful. These are the short term goals that we wanted to have. And maybe for longer term goals, the rehab component will be a project that can be done for a few years in the future. Maybe that's the way we go.
So Jim, do you want to read out that question, Jim?
Can I follow the question?
Oh, yes, Ma'am AKR, please.
Yeah, I actually posed a question for Dr. Rene. I asked him why there's so many accumulators that are available, yet the companies are not using them. I don't know how this is an issue. You know, we were into the bioremediation group 10 years ago. It still is the issue, and mining companies should be funding all these rehabilitation activities. The other issue is what to do with the biomass. Again, it was discussed before that maybe incineration could be an approach. Right now in Congress, there is a bill which is being sponsored to lift the ban for incineration. I attended the hearing, but there were so many against it. They said that incineration, you have to put filters to avoid the release of toxic compounds in the environment, but that is very expensive and may not be applicable to the Philippines, they said. So I don't know, what will we do with the biomass once the heavy metals are accumulated? Still an issue. A decade ago, still an issue. What do you think, Rene?
Thank you, Ma'am Raimundo. Actually, we did some experimental studies on recovering the metal from the plants, particularly copper and arsenic. We are not familiar with nickel for now. Before, we thought of incineration as a process, but the other was Fisheries actually discouraged that method, so they look into the wet analysis or extraction. So the use of acids. So that's what we are looking now. So we can actually extract copper using nitric acid, digesting it with nitric acid, and also with arsenic. So it can be done, ma'am. It can be done by not burning. For nickel, I am not so sure on how the process would be. So if there's a support on a bigger scale, we want to experiment on this on a bigger scale. Maybe it can be a possible or feasible way of extracting metals from the plants. So there's a way, ma'am. There's a way. So I think we're still hooked in doing this. There's another question you asked earlier. Why are not the mining companies using these hyperaccumulators in revegetating their abandoned mines? Actually, we did an experiment on that at Manila Mining. As I mentioned earlier in my presentation, it was the plant care project that was a challenge to us. It is quite difficult to transplant because it should be a personal, there should be some personal touch maybe that can be inputted for plants to grow. Otherwise, I think more than 50% of your hyperaccumulators would eventually die. That's a challenge, man, for us.
You need a botanist in the group.
Yeah, you need a botanist for that. Yeah.
Okay, thank you.
Actually, can I add to that question? Because what Ma'am AKR says is a very valid point. But another thing that we have to think beyond the box is these plants are really remarkable because they can withstand such serious insult, if you like, physiological insult. Therefore, we have to try to explore what are the metabolomic processes that are occurring, because if we are able to understand that, we can even think that these can be possibly used for medicinal purposes. For example, the French have started doing some research on the enzymatic processes in some of the nickel hyperaccumulators of New Caledonia. And they have already patented some of these, processes, because they realize that these are in fact, they are marketing it as eco-nickel or bio-nickel, because you can actually use those enzymatic processes for other feedstock, for other processes which can be much more valuable. So you might only need, let's say, 1 kilo of leaves from a nickel hyperaccumulator to create, let's say, glutathione, which is an important antioxidant. So this really asks the question, we have the capacity in the Philippines with our biochemists, chemical engineers, to really try to understand, unpack these processes to produce something which is probably so much more value adding.
Okay, thank you.
And I think there is a question here from Amelita Luna for Dr. Claveria. Are there any shrubs or small trees that can absorb high concentrations of both copper and nickel, copper and arsenic, other than local ferns?
Unfortunately, we did not encounter larger plants, I would say, or shrubs that can actually hyperaccumulate copper and arsenic. We tested a few, especially those metallophytes, the general metallophytes, that they grow also in toxic areas. Although they don't accumulate any of these metals, they just grow there and survive. But they do not absorb most of these metals, so we don't use them as our examples or species for experiments. So I'm not sure. Maybe you can help us identify some of these larger plants that can accumulate copper and arsenic.
Yes, copper is a particularly difficult metal to deal with because it is very toxic to living tissue. But I think the Chinese have been doing some work where they have actually some mass planting of some copper hyperaccumulators, and if you're interested, I'll send you the references of the work. They're mainly from South China, so the same type of climatic conditions as us. Now, I think there is a question here from Dr. Leticia Afwang. So Jem, can you say what Dr. Afwang's question is?
So her question, this was posted on Facebook. She was asking, do we have data on the extent or impact of remediation on mining wasted ecosystems?
I think Imam AKR can answer that. She's been a champion to try to make sure that these places are actually remediated.
What was that? Dr. Afwang was asking about the impact.
So she was asking, do we have any data on the extent or impact of remediation on mining wasted ecosystems?
I think there are there are studies on this, and just the protracted the study of Carlo, of Dr. David of UP Diliman. They have really studied the impact of the mining industry in Bicol. There are available data, but I don't have it on hand now. But of course, it's affecting the environment. Yeah. So Letty, just let me know. We'll get together at IBS for that.
Yes. Okay. Thank you very much. Well, I think we'll unfortunately have to draw proceedings to a close. It's been a wonderful session, the both sessions. I'm really appreciative of the speakers in really sharing something of their passion in the research that they have done. And I think you can all see that there is an application in all this. And really, as you know, PAASE's outreach is really to make it relevant. And really, when you think that in this situation of lockdown, it really has provided us the opportunity to concentrate our efforts in really providing a voice and saying what we should really use these hard-earned skills in many ways for the betterment of our Filipino people and our country. So without much ado, I would like to thank everyone, and perhaps, Jem, we could coordinate one more group photo before we go, and then we'll leave it at that.
Yes, let's do that right now. I'll just switch everyone to gallery mode again, and everyone can turn on their videos so that we can get a picture. And we want to say hello to the people who are watching on Facebook right now. There's 37 of them. Kanina, we reached up to like, I think 60 plus. So thank you for also viewing this on Facebook. All right, so does everyone have their, I think some people still don't have their videos on.
Tina is there, yes. Amelita, Luna, and Nina. Vicky Migo.
All right.
We're almost there.
Okay, wait a minute. All right.
Okay, there's Nina.
Wait
yes,
for some people
yes.
to turn on their.
I like your background, Nina.
Thank you, Yob.
All right, I guess we can do it now. I'll just press screen, print screen. Hi, everyone, smile. 1, 2, 3. All right.
Thank you.
Okay.
Thank you.
Siguro before we end, I'll also just play the video for PAASE as an ending. But thank you, guys, for coming.
Okay,
thank you. Thank you, everybody.
Thank you very much.
Bye.
Bye. Bye, Demi.
Bye.
Oh, ma'am. Thank you. Bye-bye.
Yes, I Nina.
Bye-bye.
Oh,
Thank you.
see you sa July 27.
Yes, I look forward.
Inviting all of you.
Okay, bye. Thank you, Yob.
Thank you. Thank you. I'll send you all an email.
Renee. Okay.
Bye.
Mom,
Thank you. Thank you, everybody.
So, Jem, how do we save the chat? Could you save it for me? Because I don't know how to.
Yeah, I can save it.
So I can for the report. Thank you. Maraming salamat. You know, this was a wonderful effort. It's Joel. Yes.
Bye, everyone.
Bye. I'll get in touch with everyone. Yes.
Get in touch.
Thank you very much, Jim. I think we were able to do something very, very important.